Kote, the country director for AFD. Please join us. I hope we have enough chairs. Yeah. I think it's enough, right? Okay, so I think we're set. So this morning I'm going to be co-emceeing with my friend, Professor Nikes Ndam, who is our collaborator and one of the RRD lead persons in Ghana and now in Benin. So Nikes would also be helping steer this morning's program. So I would like to acknowledge the presence of some colleagues. Professor Mark Yidana is the Dean of the School of Physical and Mathematical Sciences. Okay, our Pro-VC is here, Pro-Vice Chancellor for Research, Innovation, and Development, who's here to represent the Vice Chancellor. Please come upstage. Yes. So in the University of Ghana, we have two Pro-Vice Chancellors, so that you don't get confused. So, yeah, I'll sit here. So, he's my colleague. He's the Pro-Vice Chancellor responsible for Research, Innovation, and Development. And I'm Pro-Vice Chancellor responsible for Academic and Student Affairs. So he's here to represent the Vice Chancellor, who I said is out of town. This morning, we are joined by our colleagues from the Creek Africa Network, led by Dr. Jean Langhorn. Jean is over there, and they are here with a very strong team from the Francis Crick Institute. And they'll be joining us for the next few days. And at the end of the program, we'll have some discussions about collaborating between Francophone and Anglophone countries. So, ladies and gentlemen, it's my pleasure to introduce you to WANIDA. So WANIDA stands for the West African Network of Infectious Disease, ACEs. ACEs stands for African Centers of Excellence. So the African Centers of Excellence, as you know, is a program that was initiated by the World Bank as part of their efforts to build scientific capacity on the African continent. And the main goal of the project was to provide specialized training through the establishment of specialized centers of excellence. And WACCBIP, where we sit today, where I'm the founding director, was one of the first beneficiaries of the ACE project. Subsequently, there was a phase 2 and phase 3, and as part of the phase 3, which is called ACE Impact, there was a partnership with RRD and AFD to establish what we call ACE Partner, which is a partnership network aimed at fostering further partnerships between African institutions. And so as part of that program, we put in a proposal to establish WANIDA. And so WANIDA is part of ACE Partner, which is built on the ACE project. So you're going to be hearing a lot of ACE. ACE stands for African Centers of Excellence. So WANIDA had 7 founding partners. I would mention their names, and we welcome their teams that are here. Each of them are represented here by a team of scientists. So, of course, WACCBIP is the host. So, WACCBIP, where are the WACCBIP reps? Including myself, WACCBIP, please wave. Then we have ACEGID, African Center of Excellence for Genomics of Infectious Diseases in Nigeria. ACEGID, we have ACEGID here. ACEGID is over there. Welcome. We have African Center of Excellence for Mycotoxin and Food Safety. This is also from Nigeria. Then another one from Nigeria, African Center of Excellence for Neglected Tropical Disease and Forensic Biology. Yeah? Strong team. Then from Burkina Faso, we have iTech MTV, which is based in Bobo Dioulasso, ITech MTV, and then from Guinea, we have the African Center of Excellence for Prevention and Control of Disease Transmission, which is CPCMT. Our friends from Guinea, yeah? All right, great. So we have. Oh, did we miss Kanu? SFAB, we miss SFAB. SFAB is one of our most active centers, sorry. SFAB is from Bayero University, yeah? So African Center of Excellence for Population Health and Policy, SFAB. Yeah. Professor Maman, did I mention you? Okay. Okay, I didn't forget you. Good. All right. So we have representation from the Association of African Universities, World Bank, GTEC. Edmond is here on behalf of GTEC. Dr. Edmond. I don't want to try your name again. Yeah, but Edmond is from GTEC and is the project coordinator for the national level for the ACE project. So, have I left anybody out? So, everybody is very welcome to WACCBIP, very welcome to University of Ghana, and welcome to this conference. And we look forward to having a good time all together in the next 3 days. Nikes, please come and fill in the gaps if I left some.
Yeah, just to fill with 2 new centers that joined the WANIDA.
Yeah, go ahead.
Yeah, these are IRCB from Benin. I think they are there. And Sermes from Niger. Ronan is there.
Great. Good. So for WANIDA, even though we have the 7 founding members, because we want to build partnerships across the continent, we open it up for institutions or centers or partners to join. And so you need to just express interest to join. And the WANIDA partners would consider what value you bring and what value we can give you, and then we accept you into the network. So the last 2 that Nikes introduced are our new members who have joined us, in addition to the 7 founding members. All right, thank you. So we would go ahead and take some short remarks from our guest. So our landlady, the Vice Chancellor, is not in town. So she's being represented by Professor Felix Asante, who's our Pro-Vice Chancellor responsible for research, innovation, and development. So he would welcome all of us with some brief remarks. Thank you.
Good morning, colleagues. On behalf of the Vice Chancellor, who would have wished to be here, but she's currently out of the country. And on behalf of the management of the University of Ghana, I wish to welcome you all to this network symposium. I've been following as responsible for research. I know when this network was formed because I know we had to issue sub-awards and contracts to member institutions. And I'm happy that WACCBIP is hosting this symposium. The topic, infectious disease in an era of global change, innovative multidisciplinary Pan-African efforts towards building research capacity in Africa. This is a mouthful. I know it's only the scientists who can say this with ease. I'm a social scientist. That's why I found it very difficult. But I think the topic is very, very timely, and I must say the whole area of infectious disease is a priority area for University of Ghana. We are currently working on our new research strategy, and infectious disease and communicable diseases features very, very, very prominently. We encourage multi and transdisciplinary research, and I'm hoping and I know Professor Wandari is encouraging that in this symposium, and also how you tackle research topics in this area. So once again, on behalf of the university, I wish to welcome you all. Please find time and visit our beautiful campus. And if you have any questions on why the university is this, Professor Wandari will answer it for you. So once again, welcome, and please, we are at your service, and we are happy that you are here, and enjoy your stay at the University of Ghana. Thank you.
Thank you very much, Felix. Please, Ekua online would quickly take her remarks if she's online. Is she there? Okay, all right. So it's my pleasure to introduce Dr. Ekua Bento. Ekua Bento is a senior education specialist at the World Bank and the task team leader. These are terminologies from the World Bank. Task team leader basically means the person who's responsible for the World Bank ACE Impact project. So she's all of us our boss. And she's also from Ghana. So we're proud to have a Ghanaian holding such a significant position at the World Bank. So Ekua, please, your remarks. Thank you.
Today, the world is dealing with several challenges that know no boundaries and therefore require efforts from across the globe. Africa, which bears the brunt of many infectious and other types of diseases, can therefore not be left behind in these efforts and must be part of the solutions, and not only as a testing bed, but as part of the scientific engine that drives research behind these solutions. It is my hope that we are able to go beyond the lifetime of the AIDS project in terms of the strong scientific collaboration that is being built within these networks. More and more, I have been receiving a lot of requests for partnerships on ACEs, and I frequently say that the ACE centers always make my work so much easier because we no longer have to struggle to make the case that our ACEs are making strides in delivering on innovative and quality research. So thank you. I wish you the very best in your deliberations during this symposium. And hopefully I'll be able to join you again during the closing, but I have other engagement. I'll try my best, but I wish you the very best for the next couple of days as you come together and discuss your research and form various different partnerships. Thank you.
Thank you very much, Ekuya. I think I missed to introduce one of our friends who's joining us from Harvard, Dr. Jacques Kodbonu, who's also a Ghanaian and who has been engaging with WANIDA, trying to build partnerships. Yeah, and Gregory needs to also be introduced. Gregory is the project manager for, project coordinator for the ACE Partner Project. And Olivia, who's sitting next to him, is the project manager for WANIDA, who put all this together, who organized all this. So, Olivia, please take your bow. All right, so we'll move forward. We'll get some remarks from Florent. Your turn? Okay, thanks.
Okay, ladies and gentlemen, dear colleagues and friends, all protocol observed. On behalf of His Excellency Jules Armand Amiambossou, Ambassador of France to Ghana, it's my pleasure to welcome you in Accra on the impressive premises of WACCBIP, so the West African Center for Cell Biology of Infectious Pathogens of the University of Ghana. His Excellency asked me to convey his sincere apologies for not being able to make it this morning due to previous commitments. I know that he's involved notably in several events linked to the International Women's Day, which takes place today. So this gives me the opportunity to wish all women present in the audience a very good Women's Day. This also, thank you. Gordon and Nikkei, this also gives me the opportunity to congratulate the symposium organizers, since I've seen in the program that many presentations will be made by women. So this is clearly the way forward. So I'm very happy to be here with you this morning for numerous reasons, both professional and personal. Because of time constraints, of course, I'll mention only a few. Firstly, this symposium is fully in line with the priorities set by President Macron at several occasions, and most recently during the press conference he held on the 27th of February on partnerships with Africa. He mentioned indeed that education, professional training, employment, health are, among others, priority topics for collaborations between France and African countries. Another reason is obviously the very high quality of research and training performed through WANIDA since its inception, with, I've been told, 19 masters and PhD students already trained in members' laboratories, the participation of WANIDA faculty in 36 international symposia and workshops, and the publication of more than 15 scientific papers co-published by WANIDA members, and of course with several more in the pipeline. Yet another positive aspect of WANIDA is its regional dimension, provided by the participation, as has been told already, participation of African centers of excellence from Anglophone countries, 4 ACs from Nigeria, 1 from Ghana, who is hosting us this week, and from Francophone countries, 1 from Burkina Faso and 1 from Guinea. Another good news is that WANIDA has expanded in 2022 with the integration of other key partners in West Africa, the IRCB, Institut de Recherche Clinique du Bénin, and the CERMES, Centre de Recherche Médicale et Sanitaire from Niger. One last point I would like to underline from a personal perspective is the strong involvement of French institutions, first the AFD, the French Development Agency, through the funding provided to the ACE partner program. But my colleague Christophe Coté, who is here with us this morning, will certainly address this issue during his coming speech. The second French institution I would like to acknowledge is the IRD, the French National Research Institute for Sustainable Development, for efficiently coordinating this ACE partner program. It should be noted also that INRIA, the National Institute for Research in Digital Science and Technology, contributes both to ACE partner and to WANIDA, in the latter case through the provision of artificial intelligence tools in the framework of a PhD project. I would also like to mention the strong support provided by the IRD researchers who are posted in the countries which participate in WANIDA, and notably the IRD team in Benin, and also particularly the IRD researchers who are present here in Ghana. Indeed, they have been instrumental in developing excellent collaborations with Ghanaian universities and research institutes. So they are hosted since 2015 on the UG campus at the Noguchi Memorial Institute for Medical Research. And I would like to take this opportunity to thank Boss Prof. Nana Abba Amfo, Vice Chancellor of UG. Please convey the thanks of the French Embassy to her. And also Professor Dorothy Yeboamanu, Director of the Noguchi Institute, for their continuous support, which makes this collaboration between Ghanaian and French research team a success. Before concluding this speech, I'd like to mention a more personal point. Before joining the French Embassy in Ghana, as attaché for scientific and higher education cooperation 1 year ago, I've had the privilege to work for 4 years as the representative of IRD in Benin. So you will easily understand that I am particularly happy to welcome the delegation from Benin this morning. So dear friends, it is really a great pleasure to meet you again. In conclusion, on behalf of His Excellency Jules Armand Niambosu, Ambassador of France to Ghana, I wish you all a very good stay in this country and a very successful symposium, and I thank you for your attention.
Thank you. Thank you very much, Florent. So we would like to take some remarks also from the country director for AFD, Dr. Christophe Coté.
Thank you. Dear participants, dear colleagues, dear friends, it's both a pleasure and an honor to be here today for the opening of this 3-day symposium. And I would like first to thank the WACCBIP for hosting us today. When AFD is for Agence Française de Développement, French Development Agency, decided in 2019 to support the ACE partner project with a grant of 6 million euros, I cannot understate how much convinced we were that it was important to have the WANIDA network in place to encourage greater cooperation on infectious disease between West African countries and West African research centers. But I must admit that the achievements of the project have gone beyond our expectations. I think we can. With the COVID-19 crisis, Center of Excellence in Health participating in the ACE Impact Program and the WANIDA network have been at the forefront of the emergency response. As just one example, and I think it's a good place to mention that, at the very beginning of the Ghana outbreak of the COVID-19, WACCBIP helped develop very rapidly cheap diagnostic methods which allowed for detections and diagnostics of the COVID virus. Thank you for that. The ACE Partner Project is an institutional cooperation between the World Bank, the Association of African Universities, the French Development Agency, AFD, the IRD, and INRIA. It aims to promote the outreach and collaboration of thematic research networks between African Centers of Excellence, ACE, the key actors in quality education and research mobilized around national and regional development issues. WANIDA network aims at developing mechanisms for sharing resources between West African researchers and technical expertise. This high-level symposium showcases that wish, I think, which is to bring together renewed experts and practitioners to discuss research and its impact on crucial issues for Africa, such as surveillance, preparedness for emerging pandemics, disease prevention strategies, and vaccinology, neglected tropical disease, etc. Let me also take the opportunity of the event to say that AFD supported 2 other projects in partnership with the WACCBIP. In 2020, the IACOF project, coordinated by IRD, financed seroprevalence studies in the Greater Accra region to analyze the dynamics of SARS-CoV-2 infection. It highlighted the higher than anticipated level of prevalence of the virus in the population, in the Ghanaian population. 13.8% in November, 40% in July 2021. This project has been prolonged by the AfroScreen project to enhance genomic sequencing capacities of 13 African countries. Let me take this opportunity to salute the quality of this research partnership between internationally leading researchers playing a key role in supporting evidence-based decision-making. To conclude, thank you, WACCBIP, ACE partner teams, and let me thank especially Olivia Koupaki, coordinator of the WANIDA network, for its hard work in setting up such a high-level event. We all look forward for participating to the plenary and keynote sessions, and we are confident that this event will bring further collaboration at a Pan-African scale. Thank you.
I would like to crave your indulgence to draw attention to a technical issue with the interpretation device. So we have 2 devices. One is Bosch. So if you are using the Bosch device, Channel 1 is for English and channel 2 is for French. And if you are using the Braille device, channel 2 is for English and channel 3 is for French. So please take note. Thank you.
Okay, good. Thank you. Thank you very much, Christophe.
You should say that in French also. I can do that for you.
Okay, please help us. I was going to try, but it's okay. I'll let you.
Juste une considération technique. L'appareil de traduction simultanée, le canal 1 est pour l'anglais et le canal 2 est pour le français. Merci beaucoup.
Merci beaucoup. All right. So we're going to take remarks from our guest of honor shortly, but continuing with my introductions, I want to introduce David, who is currently the RD resident officer here at the University of Ghana. David. I also forgot to introduce my colleague, Dr. Peter Kwashi, who is a senior research fellow here and deputy director for research at WACCBIP. And we are joined by our head of department for chemistry, Dr. Mary Chama. Okay, so it's my pleasure now to introduce our guest of honor, the Secretary General of the Association of African Universities. The AAU is where the World Bank ACE project is hosted. And so he is also one of our big bosses. So my pleasure to welcome Professor Olusola Oyewole, Prof.
Good morning, ladies and gentlemen. I bring you greetings from the Association of African Universities and permit me to also join others in congratulating our women on the celebration of the Women's Week at this particular time. Congratulations to all our women. I'm greatly honored to be part of this very important meeting, which happens to be the maiden symposium of the West African Network of Centers of Excellence on Infectious Diseases, WANIDA, seeking to bolster Africa's research capacity in tackling infectious diseases in the region. Ladies and gentlemen, we recognize the wind of global change that has ushered the world into an unprecedented era of technological, demographic, and climate transformations, as well as an increased risk of infectious disease outbreaks. We all will agree that the 21st century has witnessed a wave of severe infectious disease outbreaks, which has a devastating impact on lives and livelihoods around the globe. Africa has had its share of diseases peculiar to the region, including the Ebola virus, the Lassa fever, Zika virus, among others, which resulted in substantial morbidity and mortality in multiple African countries. This has necessitated concerted efforts to build capacities in combating infectious diseases in the region. The need to provide African solutions to African problems has become key to Africa's sustainable development and growth. I should mention that one of the lessons that COVID-19 had taught us in Africa is the need for African governments and African institutions to build their own capacities to meet the challenges of their time. Ladies and gentlemen, considerable efforts in scientific research and innovations have been made as contributions to global efforts to reduce the burden and threat of locally prevalent infectious diseases in Africa. The African Higher Education Centers of Excellence project is a supporting force in strengthening the capacity of African universities to address the developmental challenges in priority areas which include infectious diseases. I'm glad to note that very exciting outcomes have been recorded from the project supports in this regard, and this could be attributed to almost all of us present at this meeting today. I must commend the World Bank, the AFD, IRD, and of course the AU in the implementation of the ACE Partner Project, which has initiated 4 networks in demanding priority areas such as infectious diseases, mining and sustainable environment, sanitation and water, power and energy, and has also led to the development of other 8 networks funded by the World Bank ACE priority in the World Bank ACE priority areas. These networks have fostered inter-ACE collaborations in addressing developmental challenges unique to the African region. And I believe that this partnership will provide some and other networks with aligned visions, just as we've seen in WANIDA. I would like to emphasize that to enhance research excellence and strengthen research capabilities, the need for regional collaboration and adopting innovative multidisciplinary approaches cannot be underestimated. Ladies and gentlemen, apart from funding opportunities, networking platforms that promote regional collaborations among researchers remain very crucial. It is in this line that at AAU, just in December, we initiated what we call the African Research and Innovation Development Networks, AFRID. Our intentions in raising up AFRID is to see how we can bring African researchers working in the same area in the whole of the continent to work together. One of the other things we are doing in the AFRID network is that we are challenging universities to recognize their areas of strength and brand themselves in these areas of strength. We hope that with this, we'll be helping universities in Africa to use research to contribute to the development of their own region. It is my belief, ladies and gentlemen, that for Africa to move forward, for us to survive at such a time as this, let us work together. Collaboration is very important, and we need to network with one another. And I believe that these are the focus of WANIDA. Let me conclude by appreciating in absentia the Vice Chancellor of the University of Ghana, Professor Amfo, and the WACCBIP Director, Professor Wandare, for hosting such an important meeting. Further thanks to all stakeholders and centers gathered here today. I'm hopeful that the discussions to be held this week will be the genesis of greater works in the infectious disease landscape. Before I leave, I'll be glad to see many of you visiting us at the AAU. You are welcome, and to let you know
Thank you very much, Prof. Prof has summed it up well. We need to work together, and that's what WANIDA is about. So we would conclude this session of remarks, and then after that we'll take the keynote address or the keynote lecture from our colleague from the NIH, and then after that there will be a family photo outside and coffee break. So in that order. So we run a little bit behind time, but we'll catch up. At this point, I think that for the sake of the presentation, we would probably have to sit this way to watch Patrick's presentation. Whilst we move, I will call on Nikes to introduce our distinguished keynote speaker. Those of us who are students of malaria research, we've watched and admired Dr. Duffy for many, many years. And so at WACCBIP, we're really honored that he made the time to join us here. And we look forward to having good discussions about collaborations with him. He's also currently hosting one of our young research fellows, Dr. Henrietta Mensah Brown. Hetty is doing her NIH APTIP postdoc with Patrick, and I'm hearing that they are doing great things together. So hopefully it forms a foundation for Patrick to return to Ghana and work here. So thank you. Nikes will do the formal introduction. Thank you very much.
The representative of the institutions, founders from the embassy, French embassy, and AFD, dear colleagues, dear friends, all protocol observed. After this first part of the official introduction of the WANIDA Symposium, and I have the privilege to introduce the first keynote speakers, so Professor Patrick Duffy. And before that, I will have to acknowledge the presence here of Professor Koita from Mali, Professor Richard Njouom from Pasteur Cameroon, Professor Sami Amidou from Venda University, South Africa, and Dr. Claude Evray from Merck, and Jane from Crick Institute Network. So I have the privilege to introduce the first keynote speaker, Patrick Duffy. I knew Patrick through his work on placenta malaria, his work on Michael Fried, who were the first to elucidate the molecular mechanism underlying the pathogenesis of placenta malaria. So Pat and Michael were the first to identify the receptors, chondroitin sulfate A, that mediates the placenta tropism of infected red blood cells. And this discovery impacted the work of many researchers, including mine, and they put us on the track of the vaccine research aimed at protecting the sensitive organs of the host. So this is at the base of emerging vaccines on placenta malaria, and Patrick is the chief of the Laboratory of Malaria Immunology and Vaccinology at NIH. And he is also, as Gordon was saying, he is a pioneer. He has pioneered a path in partnership with Mali colleagues to test most of promising malaria vaccine in pregnant women that they are developing. And I will therefore leave the floor to Pat Duffy to take us through the topic of malaria pathogenesis and vaccine discovery. Patrick.
Okay, well, I want to thank Nikes and Gordon for the invitation to join. It's really impressive to see what WANIDA is, and that was one of the encouragements that Nikes gave me, was to come and see this amazing network of scientific groups across West Africa. We've been heavily involved in training ourselves of US scientists, but also many African scientists, so I'm really inspired what everybody's doing, including the funders who provide the resources to make all of this possible. So Nikes and Gordon asked me to talk about how pathogenesis research is related to translating discovery into products for people. So that's what I'm going to talk about today. Now, how do I control this? This one? Yes. So I'll quickly talk about malaria epidemiology and disease burden and how that informs studies of pathogenesis, including host-parasite interactions. I'm going to focus on what Nikes talked about, which is pregnancy malaria, and how research over the past 30 years puts us on the cusp, we believe, of a vaccine that can protect pregnant women, their unborn children, and their newborns from disease and death due to pregnancy malaria. And then finally, I'm going to touch on severe malaria epidemiology and how that informs pathogenesis and how we should be thinking about a vaccine for children also. So let me start with malaria epidemiology and disease burden. Malaria, for those of you who are not working on it, has some distinctive epidemiological features which really tell us something about what the underlying disease is. This works. So in general, during the earliest months of life, you're protected, but then there's a pretty rapid increase in the incidence and prevalence of infection. And this really lasts for many years until about adolescence, when there's gradually an improving control of parasitemia that you measure by taking blood smears from people. And then you get to an adult level of protection where you continue to be infected, but you don't get sick, and it's not infected as frequently. There's a very distinctive feature, which is it's severe malaria, which actually occurs during a relatively brief window of life. So if your child lives to be 5 years old in many of these communities, they're not going to die from malaria. And that is responsible for 500,000 or more deaths each year of primarily African children. That looks like a vaccine because it is controlled relatively quickly, but because like other adults, they control malaria. But when they have their first pregnancy, there's suddenly a renewed susceptibility to infection. And then that susceptibility appears to decrease over successive pregnancies. And when myself and my colleague, Mikhail Fried, that Nikes mentioned, entered this field back in the early 90s, it was thought that pregnant women became susceptible because they were immunosuppressed or immunomodulated during pregnancy because the fetus is a foreign entity. But that wouldn't explain why control would improve over successive pregnancies. So it puzzled us and it inspired us to work with colleagues to study this problem. I guess I have to do that. Here we go. So first of all, to recognize that this is a problem that is not captured in WHO numbers. So when WHO tells you that there's 600 and something thousand malaria deaths every year, they're not including the deaths that are related to many of the deaths that are related to pregnancy malaria because it's difficult to see those. In high transmission areas, women that become pregnant have systemic immunity that prevents them from having many symptoms. They nevertheless have an insidious development of severe malarial anemia that it can contribute to deaths around the time of delivery due to hemorrhage. And then we also provided evidence that gestational hypertension. Thank you. Is another aspect that I'm not going to be talking about. For the developing fetus and the newborn, the most measurable and first identified problem was low birth weight in the offspring and preterm delivery that predisposed them to mortality during infancy. And in studies that have looked at chemoprophylaxis studies, about a third of perinatal mortality is related to malaria in these communities. Let's see my picture appear down there. In low transmission areas, women don't have systemic immunity. So actually what you see in those populations is something that looks like what happens in children. They develop severe syndromes and they're at higher risk for cerebral malaria, respiratory distress, and acute severe malarial anemia than other members of the population, non-pregnant women. And there's been recent evidence in these populations that they also suffer from gestational hypertension or preeclampsia related to Plasmodium falciparum malaria. But for the offspring, you see similar sequelae, which is low birth weight, abortion and preterm delivery, and neonatal and infant mortality that is clearer to see because the women are having severe episodes themselves. So you can relate those poor outcomes for the offspring to the actual malaria case that the woman's having. The estimates are about 50 million women in areas of high transmission area are exposed every year and 75 million in low transmission areas, but the greatest burden is in these high transmission areas just because of the frequency of malaria in those populations. And in Africa, the recent estimates are there are up to 50,000 women who die, and there are 200,000 stillbirths. And I mentioned to you that also low birth weight and preterm delivery are predisposing the offspring to neonatal and infant mortality. That's hard to measure because the women didn't feel very sick. So it's a heavy burden of disease, and it's really not captured by those numbers from the WHO. So we actually have been, this is my colleague Mikhail Fried, who I've been working on this problem for 30 years. We started this work as part of the Kenya Medical Research Institute, and now we have a strong partnership with our colleagues in Mali, and specifically Professor Alassane Diko is leading these studies. But one of the things we've done is to try to improve the ways that we can capture information about women's malaria episodes and tie them more closely to the poor outcomes for women and offspring. And we do this by intensively following women for malaria episodes. And what's important to recognize here is that Mali is a place where antimalarial drugs still work, sulfadoxine, pyrimethamine, and bed nets work, and they're pretty well applied. Despite that, we can see that most of the women in our populations that we follow have malaria during pregnancy, despite the application of the available tools that are recommended. And because of its frequency and the intensity of our studies, we have been able to show that malaria infection is common and associated with perinatal and perinatal mortality and preterm delivery, despite the fact that these women were getting the measures that are recommended for them. And what we also showed is that this relationship between malaria and poor outcomes is related to systemic inflammatory responses during malaria episodes that appear to be associated with these poor pregnancy outcomes. And this is the kind of evidence. This is actually evidence that Mikhail and I developed when we were working as part of the Kenya Medical Research Institute, showing that in children that are born low birth weight, as an example, TNF-alpha levels are significantly higher in the placentas of those women than children that are born normal birth weight in an area where women are being exposed. So there's a relationship between infection, inflammation, and the poor outcomes, both for the women as well as their offspring that I was telling you about. So just to describe pregnancy malaria to those in the audience that are not familiar, it has this distinct parity-specific epidemiology of risk during first pregnancy that decreases over sequential gestations. Pregnancy malaria syndrome and presentation depends on the state of the mother's maternal immunity, the amount of anti-malaria immunity that she has acquired over her life. But in both low transmission areas and high transmission areas, even if you have systemic immunity, pregnancy malaria is causing maternal, fetal, and newborn mortality, and that is related to placental inflammation. So now let me talk to you about, with that framework, how studies of pathogenesis have informed the effort to develop interventions. So this is what placenta malaria looks like. It's called placenta malaria because the parasites accumulate in the intervillous spaces. This has been recognized since about 1900, not long after Laveran discovered the malaria parasite in Algeria. So what you can see here are mature forms of parasites that are accumulating in these intervillous spaces. And actually, the work that Mikhail and I did speculated that this must be a specific binding event and that this was a distinct parasite phenotype, and that's essentially what we found out. We work with Dr. Alex Masori at the Nyanza Regional Hospital in Kisumu, and enrolled women who were just coming in for their deliveries and who would donate their placentas so we could study the parasites that were infecting the placentas. And what we found really quickly within a few weeks was that the parasites had this property of binding to a section of a placenta that's not infected. So we would make cryosections of uninfected placentas, allow parasites to settle onto the tissue and wash away what didn't bind. And what you can see is what we found was that parasites would bind right along the surface of the villus that's in the placenta. That's the fetal tissue in the placenta, but they're binding in at where there would be an interface with the maternal vascular space. So this would be where the maternal blood had access. And this was later studies when we realized what it was binding to. Nikes already mentioned to you chondroitin sulfate A. So if we treat that placental section with chondroitinase to remove this glycan called chondroitin sulfate A, the parasitized red cells no longer bind there. And so pregnancy parasites bind to chondroitin sulfate A, and that distinguishes them from parasites that you collect from other people. So this is just an example of 2 parasites we had from placentas, 2 that we had from males. CD36 is a very common receptor that supports the binding of parasites. We have many more data, other groups have data, that if you collect parasites from placentas, they uniformly bind to this chondroitin sulfate A, and it's a very uncommon phenotype in other populations. So what happens is during pregnancy, you get a new organ, it has a different receptor, and it selects a distinct parasite phenotype to overgrow in the placenta. And so CSA binding parasites sort of became a shorthand for the parasites that would infect women. And I don't want to go through this data in great detail, but essentially what we found out, what this shows is the level of parasite binding that are Kenyan parasites that we collected from pregnant Kenyan women, and looked to see whether sera from first-time mothers, second-time mothers, or third-time mothers would reduce that binding to this placental receptor chondroitin sulfate A. And what you can see is indeed sera that we collected from women in Kenya. First-time mothers did not inhibit the binding, whereas the multigravid women, their sera would inhibit the binding. We also found the same phenomenon with parasites from Malawi and parasites from Thailand. Women, study participants who had never been pregnant had no activity to reduce that binding. And so we found the same phenomenon when we worked with our colleagues in Thailand who provided us with parasites from Burmese women who were living in Thailand. This is Francois Nostin and his group. We found the same phenomenon with the same sera. And so what's important to recognize here is it appears that there's a very conserved process going on immunologically around the world where women are acquiring antibodies that are not only effective against the parasites that they might get in Kenya, they're also effective against the parasites that might occur in Burma. And this to me sounds like a vaccine for pregnant women, because if we can induce that, what we can do is to reduce the incidence of infection. And what I show you here is the data in Kenya showing that first of all, no primogravid women had these antibodies there, and all multigravid women had these antibodies. Only in secundigravid women that there was heterogeneity in whether they had these antibodies or not, and women who were infected during their second pregnancies were less able to control parasite binding, linking those two things. And so this is now the immunologic feature that we would want to use to reproduce with the vaccine. And so what I've told you in this section is that Plasmodium falciparum parasites sequester in the placenta, and those parasites specifically bind to a distinct receptor there, chondroitin sulfate A. Women acquire antibodies naturally that block parasites from binding to that receptor, and the pattern of those antibodies explains why women are most at risk during first pregnancies and they develop resistance over sequential pregnancies. So now let me talk about vaccine discovery for pregnancy malaria, and a very important discovery that our colleagues in Copenhagen made. This is Ali Salanti and Tortiander. And what they were doing was studying a very important variant antigen family, variant surface antigen family of Plasmodium falciparum parasites in general, called PFE and P1. There's about 60 members of that parasite family. They took a well-characterized parasite in the lab, selected it to bind to this placental receptor. All Plasmodium falciparum parasites have the potential to bind to that placental receptor. It's not a genetically distinct parasite. It just puts up a different member of this family on its surface. That's it. And that is what's called VAR2 CSA. So if you select parasites to bind to CSA, it's that single member of this highly variant family that always comes up, and it's unusual because it's the only member of that family that you can recognize as a conserved entity, conserved in the sense of recognizable in that family. So something we still don't understand the genetic basis, why it would have that and why it would bind to CSA. But nevertheless, that is the gene of interest, and that quickly became the leading target for a vaccine that Nikes and we have been working on since then. I want to briefly tell you about a new model that we've developed for this, because one of the problems with pregnancy malaria is we haven't had a good model to reproduce that specific phenomenon. So this is an Aeotis monkey, and what we have the privilege to do at NIH is to set up a breeding colonies of this in our research facilities. And so that we can get pregnant Aeotis and examine whether we can reproduce the features of human placental malaria. And so this is the basic design of the study that we started with about 10 years ago. We would pair a male and female together. At some point, the female would become pregnant. The gestation is 132 days. And when we started these studies, we were given this parasite, CS2, that is a reliable CSA binding parasite. And what we found briefly, let's see, is that I'm having some problems with that. Can you advance that? Can you go back? Yeah, you can just put that data there. So what we found briefly is that the parasites that we infected those animals with You can see the binding. These are images of the binding assay, binding to a control entity, BSA, binding to the CD36 that I mentioned, or the binding of parasites that we collected from the monkeys, the pregnant monkeys we had infected. We do get CSA binding parasites out of these monkeys. The figure that you show at the bottom shows the placental parasites that we collect from the placentas of these infected monkeys always bind 100% to the CSA. Parasites we collect from their peripheral blood. Oh. Okay. I've been asked to speak a little slower. But the peripheral blood, what you can see is sometimes you have CSA binding parasites there, and sometimes you have CD36 binding parasites there. So it's interesting. The peripheral parasites are different, not always coming from the placenta. And I just show, using antibodies, that the parasites that are in the monkey are expressing this VAR2 CSA on their surface. But this becomes a model now that we can test our vaccines with, because these are monkeys that we can reproduce the placental malaria experience in. This is the VAR2 CSA protein. It's a very difficult protein. It's very large and cysteine rich. Nobody realistically thinks we can make this as a full protein vaccine in terms of a recombinant protein. There have been entities that have been brought into the clinic that use a fragment of this. Our colleagues in Paris and in Copenhagen have brought these two vaccines called Primvac and Pamvac. We worked with them to test their vaccines in our monkeys. They were doing studies in humans and have completed those studies. And what's important to know, I won't go through all the details of this study. We would immunize, we would test the vaccine response, and then we would give the monkeys placental malaria, which is something, of course, you can't do in humans predictably. And so we could see whether there was a boosting response. But I will just sort of cut to the chase to say that the results that we got with the vaccine were very similar to the results that they got in humans, which is encouraging for the model, but not encouraging for that first generation of vaccines. What I'm showing here is sera that were raised from the two vaccines of interest, Pamvac and Primvac, blue and red. And then we had a candidate ourselves that was, I won't go into the details of that, and then a control malaria antigen. And what essentially the vaccine would induce antibodies that only recognized parasites carrying the form of R2 CSA in that parasite. But if you went to another parasite, there was no reactivity to those parasites. So it was very narrow. Did I do that? It was a very narrow level of activity. And when you looked at the functional activity, you can see the same thing, that this functional activity of blocking the parasites from binding in the placenta was limited to the parasites that carried that very specific form of R2 CSA. So this is not a vaccine that would widely protect pregnant women. And just to quickly mention that when we gave the animals placental malaria, when they had a placental malaria experience, it didn't do anything to broaden their vaccine response. It didn't provide them with broadly neutralizing antibodies. So one of the advances that we've had recently in our lab is our ability to make full-length recombinant forms of this that appear to be structurally correct. This just shows 7 different variants of VAR2 CSA on this gel. On the left side, it's non-reduced protein. On the right side, it's reduced protein. But now we have the material of VAR2 CSA to really work with and understand how immunity is working and how the protein is binding to chondroitin sulfate A. The figure on the right just shows the approach that we took to improve the expression of this protein using mammalian cells, which actually raises hope that we could make an mRNA vaccine out of VAR2 CSA. We've been able to use these for structural studies, and what I show here is structure that we obtained with our colleague Neeraj Tolya, who works in the office right next to mine. This is now the protein that's responsible for placental malaria, and what you can appreciate right here is chondroitin sulfate A actually threads its way through a pore, a channel that's created in the protein that involves actually a confluence of multiple of the domains of this very large cysteine-rich protein. And this is a schematic that shows that interface. Really, this is chondroitin sulfate A. This is a major channel that threads through, and then there appears to be a minor channel. So this large, complex protein folds around itself, creates a channel, and chondroitin sulfate A is a long chain that threads through that and allows the parasite to bind in the placenta. Some studies that one of our I guess, mid-career scientist at this point, but Justin DeRichmo, who's from Benin and worked with Nikes before he came to NIH. He used these to study how what naturally acquired protective immunity is. What I diagram here is Justin's efforts to take those multiple different variants of R2-CSA, purify antibodies on that protein, and also deplete them from the sera. And then you can use that purified antibody or depleted sera for studies that characterize the nature of those antibodies. And most importantly, this binding inhibition assay is how we test whether antibodies can block the parasites from binding to the placenta. And the important information, probably there's a whole set of data that he got, but the most important piece of information he got just by taking a single variant of R2-CSA purifying antibody and testing its ability to inhibit a wide array of placental parasites from binding to the receptor. And what you see here, it's depicted the opposite of what I previously showed you. The higher you go, the greater the activity of the sera. And what he showed is that with a single allele of R2-CSA, you purify antibodies that are reactive with parasites from around the world that infect pregnant women. So even though the vaccines that we've had go into the clinic so far were not able to induce this immunity, the full-length protein has epitopes of fully conserved antibodies. So this is very encouraging for a vaccine. So to summarize what I just told you, placental parasites, which are CSA-binding parasites, have VAR2-CSA on their surface. VAR2-CSA antibodies that block them from binding increase with parity, and that would explain why women become resistant over successive pregnancies. We have found that VAR2-CSA fragments that have been tested thus far don't induce, they induce variant-specific serum activity, but what we're looking for is broadly neutralizing antibodies. But a single full-length VAR2-CSA can purify those broadly neutralizing antibodies from the sera of multigravid women who have been exposed to malaria. And then finally, let me just touch on this, severe malaria epidemiology and what we understand about that so far, because that's another key target for malaria vaccines. This is a study that Brunner Gonçalves, a young scientist from Brazil, completed while he was in our lab, working with our colleagues in Tanzania, where we were doing studies where we enrolled children at the time of delivery. Women would come and deliver, and then we followed the children for the first 5 years of their lives. And the important findings really that came out of those studies, and it's just this one slide, is that parasite density does not explain severe malaria in children. Children don't have severe malaria because they have a high parasite density. It also showed that control of parasite density does not explain immunity to severe malaria. If you look at malaria episodes before a severe malaria episode and after, they have the same parasite densities before and after. And finally, the risk of severe malaria is constant over multiple infections in children. In other words, a children that has had 0 previous malaria experiences in an African community is at the same risk of severe malaria as a child that has had 5 or 10 episodes. You do acquire immunity over a span of years, but it's not from a mild malaria episode necessarily. So just to say that the epidemiology of severe malaria can also inform our pathogenesis studies and how we look to develop vaccines against them. This is our group at NIH, a lot of amazing scientists, and there's Michal, my colleague. And these are the leaders of the teams that are our vaccine development unit. And these are our colleagues in Mali, where we have, NIH has had a 30-year relationship with them, and it's just an amazing partnership that is truly collaborative and makes vaccine discovery and pathogenesis research move very quickly. This program, when I got there for vaccine development, started with Professor Ogobara Dombo as the lead of the vaccine trial, and he very sadly passed away a few years ago. And has been replaced in terms of the vaccine development with Dr. Asaka Sagara, one of the professors there. So I thank you for your attention. I hope that I conveyed that basic research turns into products that can help people.
Few questions? before we go to the photo. Yeah, there is a microphone in the room wall. So, Women's Day, we will start with Jane. Sorry.
Thank you, Professor, for your nice talk. Very informative for me. I'm sorry. As you remember, I'm Derrida from Burkina Faso, RSS, Institut de Recherche en Sciences de la Santé. I would like to know about altruistic vaccine. Here your talk is about placenta vaccine, but what thing I would like to know is about altruistic vaccine, transmission blocking vaccine. 10 years ago, we evaluated the efficacy of this vaccine in our lab from using the antibody from your laboratory via human. So I would like to know the issue of this vaccine.
I didn't quite catch the question.
So my question is about the issue of the altruistic vaccine, transmission blocking vaccine.
Transmission blocking
Yeah,
vaccine.
yeah, yeah.
Oh, transmission blocking vaccine. But that's
Yeah.
So there's been a lot of progress with, so transmission blocking vaccines work by targeting parasite proteins that are expressed while the parasite's in the mosquito.
Yeah.
And so what we've learned is you can vaccinate people with those proteins. They're on the surface of gametes and zygotes that only are accessible, in the mosquito. And we vaccinate people, they develop antibodies, and when the mosquito takes a blood meal, they take up those antibodies, and the antibodies attack the parasite in the mosquito.
Yeah.
And so the answer to your question is those vaccines have completed phase 2 testing,
Okay.
and they show a very high level of activity
Okay.
to prevent mosquitoes from getting infected. So the
Okay.
way we test the vaccine, this is ourselves and our colleagues in Mali, we feed mosquitoes on the vaccine trial participants on a regular basis. And so we found that it reduces infection events in mosquitoes by over 70%. Pretty consistently. So those vaccines are moving into phase 3 development. Is that what you're asking me?
Yeah.
Yeah. So those are now advancing in vaccine development.
Okay.
Happy to talk about with you some more. It's a very exciting area of vaccine
Okay, I'm
development.
interested to discuss
Yeah,
with you.
I think
Okay.
those vaccines can be combined with the R21 vaccine that was tested in Burkina Faso. Or
Yeah,
it could
yeah.
be combined with RTSS.
Yeah,
but
yeah.
they have to be given to everybody. So for those vaccines to have their effect, you have to vaccinate everybody in the community. It's herd immunity, but that's the way that they will work.
Okay, you can talk after.
Yeah, we'll talk about
Okay,
that.
thank you.
Thank you. I think there was a question in the back.
All right.
Jean, sorry.
This is going to sound stupid. I should know this. These women have antibodies presumably to other VARs. So are they immune? So if you take away the VAR2 CSA and you stop the placental malaria, is there a chance that they can get seriously ill from a non-placental malaria, or are they all immune to the subsequent VARs that would be expressed?
So actually nature has provided an experiment in a way, which is the delivery of the placenta. And what's been described is after the placenta gets delivered, a woman's parasitemia usually resolves after that is cleared. That doesn't mean that they're immune, they'll get reinfected still after pregnancy, but that the specific infection that is filled with CSA binding parasites, that will resolve as well. there's a period of susceptibility to the postpartum that's been described that's not really well understood. It could be because vascular spaces are modified during pregnancy, so maybe the chondroitin sulfate might appear in other vascular spaces, or it could be because of the blood loss during pregnancy, leads to a rebound kind of reticulocytosis, and both Plasmodium falciparum and Vivex prefer younger red cells to invade. But those women would still have all their immunity to those other PFE and P1s. And as I mentioned, if you take peripheral blood from pregnant women, lots of times there's CSA binding parasites, but lots of times there's also CD36 binding parasites, and those are obviously sequestering in other vascular beds, not the placenta. So you'll see different kinds of binding parasite phenotypes in the peripheral blood of pregnant women. So they're still getting exposed. There's somebody behind.
Yeah, thank you for the presentation. I'm asking, what is the benefit of this vaccine to the mother? And then can it be adapted in the general population, since this is a very serious problem in Africa? Thank you.
You're going to have to hold on. The acoustics here are not good. Did somebody understand the question? The acoustics are not good here. What was I'll have to ask the question again.
Okay, what is the benefit of this vaccine to the mother? Can it be protective to the mother, and then can it be adopted in the general population for other people, considering the seriousness of this disease in Africa? Can it be adapted for the general population?
I think I understand your question. This vaccine should be delivered to women before they get pregnant. Infectious agents. So I think that there should be a menu of health options for women who are entering the reproductive years. That's ideal. Part of the reason it's ideal is because women that are pregnant are getting infected at the beginning of pregnancy, and many of them don't show up until maybe the second trimester for their first antenatal visit. So exactly that you're asking. Yes, we want to be able to give it to pregnant women if they haven't gotten the vaccine, but the ideal thing would be to vaccinate women before they have their first pregnancy. That would have the greatest benefit for them. Thank you.
Thank you, Pat, for this very interesting keynote. And we'll now move to get the family photo outside. And I also I have to acknowledge the presence here of Dr. Florence Pradel from Gabriel Network. So please, let's go and meet the other officials before they leave outside. Hey, there is also...
Hello, everyone. Can you please leave your headsets behind? Your translation devices. Kindly leave them on the seat, and then when you come back, you take it. Thank you.
Ok. Juste pour nous indiquer, on peut laisser sur place, voilà, les écouteurs de traduction simultanée. Il n'y a pas de souci. Vous pouvez les laisser.
Can we all move to the front of the main biochemistry building in a timely manner so that we can take the family portrait quickly? Hello, everyone. Can we please be seated?
All right. You don't need to be so close. All right. I think let's try to get settled. We're running a few minutes behind schedule, so we have quite a packed schedule, which is a good thing. But that also means that we need to be quite tight on time. So I believe Prof. Awandari introduced me earlier. It's my pleasure, medical school. This session will be Emerging Health Challenges, Building Sustainable Systems Within the One Health Framework. All the speakers keep to time, and the chairs will make sure that that's what happens. Thank you very much.
Thank you, Dr. Kwashi. Right, so welcome to this session. Our first speaker for the plenary one is in the person of Dr. Issa Aliyu from ASPAP, Nigeria, who is going to talk on tackling SARS-CoV-2 infection in Nigeria to guide public health interventions. So Dr. Aliyu, your audience, and you have 15 minutes, please. 15 minutes presentation and 5 minutes questions and answers. Thank you.
Okay. So our protocol will you observe? We'll be presenting tracking SARS-CoV-2 infection in Nigeria to guide public health intervention. By introduction, in Nigeria, the earliest confirmed cases of COVID-19 happened on 27th February 2020 from an Italian student arriving from Milan at Murtala Muhammad International Airport in Lagos. And the active case finding of this index resulted in identifying more than 200 suspected individuals who had contact with him, and only one was happened to be positive out of those 200 suspected contacts. And though thereafter some related cases were reported, but today we have more than 267,000 reported COVID-19 cases in Nigeria. This figure is relatively very low compared to most countries, even among African countries. The relatively low number of cases in Nigeria might be probably due to the limited testing capacity across all African countries, and the fact that cases reported using QPCR in this type of test is only symptomatic subjects and or subjects who had history of contact with positive subjects are tested. So this might leave asymptomatic individuals and those who have mild disease unaccounted for. Yeah. But seroepidemiological studies can give a true estimate of COVID-19 infections by checking in the blood vaccination. Seroepidemiology can also provide critical information on the extent of COVID-19 transmission in the population, identify risk factors of infection, monitor vaccine efficacy. For this study, we aimed at tracking the transmission dynamics of SARS-CoV-2 in Nigeria to better inform public health intervention which is aimed at reducing COVID-19 associated clinical illness and mortalities. By conducting a large-scale population-based COVID-19 serological screening to detect previous or current SARS-CoV-2 infection using rapid diagnostic kits. Okay. Okay, study protocol. After obtaining the ethical approval from Federal Ministry of Health and State Ministry of Health, and having collected permission from the head of each institution who conduct the research is conducted in 4 states in Nigeria, the one in the north, 3 others from the south. But the data presented here is from the Kano state, which is the state conducted in northern part of Nigeria. And the site of studies includes the communities, shopping malls, bus stations, markets, educational institutions, and religious institutions. Okay. Having obtained the informed individuals, and then the the laboratory analysis was conducted in the field. So for the statistical analysis, we collected blood from the patient at the field. Those who happens to be IgG negative and IgM negative will be counseled, and the result will be judged. These type of individuals, they are not exposed, they are not vaccinated. So we counsel them, advise them on taking either vaccines. And then those individuals who are IgG positive and IgM negative, we also counsel them and give them the results. These are individuals who are supposedly exposed to the virus, either by infections or by vaccinations. We counsel them and give them the results. But by individuals who are IgG positive for this, they have encountered and they develop immunity against the virus. And those who are IgG negative and IgM positive, these are individuals who are recently exposed to the virus, and they might be having active infection at the moment, and they are infectious. They just swab to do rapid COVID-19 testing at the spot. So individuals who happens to be COVID-19 antigen negative will take PCR testing. Individuals who happens to be and then PCR negative will also be counseled and the result given to them. And the PCR positive, there are samples who qualifies for, this is the result interpretation I mentioned, so result and discussion. Okay. The characteristic of our patients from shopping malls, 16.6% of our subjects were from shopping malls. Oh, sorry, this is a mistake. This is the age distribution among our subjects. Individuals who age between 20 years to 39, they were 63.9% of total subjects, but at least those who are above 60 years. 3% of the total subjects we tested. For the highest level of educations, individuals who have offered secondary school have the highest number of participants in the study, while those individuals at the higher income class, which only to 5.2%. The employment status of our subjects among the subjects. Okay, so the site-specific seroprevalence. Our study shows that higher incidence of the seropositivity. This is followed by individuals from the shopping malls, with the least coming from religious institutions. And multiple logistic regressions analysis of the risk factors of positivity with the sampling site shows that it is more likely to be seropositive for COVID-19 if you are sampled at educational institutions, and then followed by shopping malls than other sites. This probably is due to the fact that these are the sites that's enclosed sites. So the chance of getting infections in this site is higher than others. Like for the communities, it's a bit higher, but it's not as high as the more enclosed site. The bus and market stations also the percentage of having positive seropositive is still higher, but it's not up to the enclosed site. So this shows probably the enclosed environments enhances or increases the chance of becoming positive for COVID-19. And this will warrant the need for enforcement of the protection measures, especially in an enclosed environment. Okay. So we also looked at the age distributions among with relation to the seropositivity, where we found that individuals at the age of 20 to 30 years of age has the highest chance of becoming seropositive. And this is because these are the most active working age in the study site. This is followed by individuals who are at the age of 40 and 50. They are also part of the working age in the society with the least frequency seen in the individuals who are above 60 years of age. Okay. Also, we looked at the seropositivity based on the gender. Also shows that it is more likely to be seropositive if you are male in Nigeria. This is because probably the males has to go out, especially in the study site, has to go out to look for the off-keeping of the family. So they are more exposed to contact with the people, and probably they have the higher chance of having COVID-19 seropositive compared to female, especially in the study site. Okay. So we also looked at the seropositivity based on the monthly incomes of our subjects, and the result shows that individuals at the low-income class this is because probably individuals at this class, even during the lockdown, they can't live at home because there is no food, and others who can actually stay at home during the lockdown. We also checked at the employment status of the subjects, where the unskilled study site, especially the markets and bus station site, these are individuals who must have to actually also, the COVID-19 protection strategies is very minimal, also eliminated, especially the bus station and lorry stations, to look for livings. So, but it's much lower in among retirees. These are individuals that happens to stay at home, especially during the lockdown. Okay. We also looked at the educational levels of the subjects, where individuals that offer secondary school levels has the highest chance of getting seropositive in our study site. This is because also they are also the workforce of secondary school level, and then the chance of them getting seropositive is high because of the type of life they found themselves. Okay. Okay. For the antigen test, we conducted antigen tests in hospital site because we want to check the level of exposure at the hospital site. If we look at the antibody, we thought that it could be higher because the higher rate of vaccines and so on and so forth in the health institutions is higher. So we tend to check the antigen, which is more likely to show the active infections. And in the site we studied, this is Multidisciplinary Specialist Hospital, where we have the 8.9% of individuals who have the antigen, COVID-19 antigen. And then this is followed by IDH, this is Infectious Diseases Hospital, also in Kano. The lowest is in Kano Teaching Hospitals. These are individuals who presented to the hospitals with clinical conditions that are similar to that of COVID-19. Most of them are in the chest clinic, dot clinic, and so on and so forth. So there are individuals who are presenting to the hospitals for other conditions, not COVID-19. But they are found to be, quite a number of them are having 67% of these individuals who presented to the hospital for. Samples who happens to be...
Dr. Liu, your time is up, please. Can you have just a minute to run up for us? Thank you.
So this is the sequencing data from the ASAF site. And then, So in conclusion, this is the first report of the large-scale population-based study for COVID-19 epidemiology in Nigeria. And we reported that COVID-19 in Nigeria, the epidemiology is varied from the gender, age, exposure site, level of education, and employment type. And the finding of this study shows that the average epidemiology of COVID-19 can be as high as 72%. This is quite much higher than what was reported using qPCR-based assays. And then a tenor of 75% level of immunity might warrant herd immunity, but this is something to actually watch because some literature say that the antibody against COVID-19 tends to disappear after a year or so. And the finding of this study further suggested that there is need to have more robust system for timely data generation and dissemination in order to guide policies and public health strategies in disease management, especially for the future management of COVID-19 and other related infectious diseases. So it's our team in the different fields. Okay, thank you very much.
Thank you. Thank you very much, Dr. Ali. Right, so we open up questions for him from the audience. Yes. Can I have the mic sent to him, please?
Thank you very much for your presentation. I noticed that you did sample over quite a long period, basically starting at a very early stage of the pandemic, December 2020, and the last samples were collected in May 2020, so just after the occurrence of the Omicron variant. Did you analyze your, one question would be, did you analyze your sample or in terms of when they were collected, how high the zero prevalence are? Because the reason I'm asking is that we had looked at one specific community in Africa and noticed that samples collected back in 21, that means before the Omicron wave, were around about what you reported, 50 to 60%. Yeah? And in the same community, samples were collected a year later, that means after occurrence of the Omicron variant, and was above 95% zero prevalence. I think it is very important, as you pointed out, to look for that. Thirdly, I would have a suggestion, a comment for rapid screening in populations for the presence of virus, because you depend on first looking at the antigen, at the antibodies. They may already have lost the virus, and until you have that, it's too late. What we did successfully just in our university to see how many people are positive at a specific site, because we want to see whether the university is a spreader, is that we collected throat washes. People are much more likely to provide throat washes, just 5 ml salt water, you gargle, and then you can pool a couple of samples and run them quickly through a reverse transcriptase with a one-step assay, and in a few hours you have an idea how high the zero prevalence is. If you're interested in that, we can talk about it later. But first of all, I would like to see whether you looked at the timing, what the frequency was.
Yeah, we actually did that. It's just that I didn't show the data. We analyzed the data before Omicron, and we also analyzed the data post-Omicron. And obviously, there wasn't a persistent pattern of like increase or decrease. The data looked somehow similar before Omicron and after Omicron. So that's why I decided to merge it. Even pre-Omicron, in some sites we had more than 67% seropositivity. At the same time, during post-Omicron, we have 70.1, 72. Somehow it's just similar, so we just took the average. But we actually have a separated time-based spatio-temporal sample analysis here. Thank you.
if I may add, because the point of you raised on providing information on herd immunity, there's also the possibility to test for blocking antibodies. Antibodies which block the binding of the spike protein to the ACE, to recombinant ACE receptor. Apparently this surrogate test works quite well and gives you a good idea on how inhibitory the antibodies are. Referring to the study which I mentioned, where we looked in the communities, we saw that in August 21, before Omicron, only high level of antibodies were sufficient to block. After the Omicron wave, even much lower titers were able to block, possibly because of reinfection. So this way you can get more of an idea of herd immunity.
Okay, thank you.
Microphone, please.
Mike?
Mike.
Maybe, thank you for this interesting talk. Maybe a provocative question. How can this, according to the data you presented, we see that the majority of the population has been in contact with the SARS-CoV-2 variants. So how can this, and this is the case in many African countries now, how, according to you, according to the result, how can this inform vaccination strategy in Africa, if there is one?
Okay. Based on the data so presented for Africa, the picture that is portrayed for Africa is that there is very, very low infection rate in Africa. And there is probably less need for vaccine, so to say, based on the data we have from the qPCR results. But obviously from what we have in this result and from the data we also produce, it shows that probably that's not the true picture of the infection in Africa. There can be large number for us to vaccinate this group of individuals. Okay. So actually the results shows that we need to do more in Africa as far as vaccine is concerned. Because the picture we used to see wasn't the actual true picture of the infection or the disease burden in Africa. Thank you.
Right, a question over there.
Sorry, excuse me.
Thank you so much for the presentation.
Okay.
2 questions. One, what do you think, what was the difference in terms of antibody and antigen detection in order to be able to see the actual infection? What was the rate of actual infection? And number 2, there is data that is coming out showing that actual infection protects more than the vaccine. So therefore, people who have been infected probably will be protected more than even taking the vaccine. So what is your opinion on that? Thank you so much.
Okay. Actually, this is what we said. I know the gold standard testing techniques for COVID-19 is RT-qPCR. But the capacity of these diagnosis techniques is less in Africa. Not only in Africa, but actually across the world. So if you are talking about the specificity, the qPCR is more specific, it's a gold standard. But affordability and then ease to perform, and even the specificity, because these are validated kits that performed more than 90% of the specificity and sensitivity. So they also gives you the true picture of whether you're positive for the virus. And it can also tell you the history. Probably you were previously exposed to the virus. You were asymptomatic, that's why you don't have any problem. But you were exposed. Large-scale testing is concerned. Antibody is the best approach to go for COVID-19. But in terms of this can be supported by qPCR testing to confirm what we are seeing. And because of the possibility of you analyzing the antigen results to classify the subject into either they having previous infections, active infections, or the recent infections. This also helps you to classify the subjects who are qualified to go further for further analysis to confirm the result. With regard to the either if you are infected naturally, you are more protected or than the vaccines. Actually, if you are infected naturally, what is seen is usually the N protein antibodies in circulations. The most vaccines, the neutralizing antibody they produce is against receptor binding domain of the spike protein of the virus. That's the antibody produced against this domain, they are neutralizing antibodies. And these are the antibodies that are generated during vaccinations. It's more likely that when you are vaccinated, the antibody that are produced, they are more specific in virus neutralizations than in the natural infection. That's my...
Right, thank you very much. There's a last question online, so can we have online participants to ask the question, please?
Dr. Kamara, we've asked you to unmute to ask your question.
Sorry, I'm not seeking to ask any question for. So my question was for the previous talk, so not that one. Sorry.
Okay, thank you.
Did you hear the question? Right, so that's okay. Thank you very much, Dr. Ali. We are grateful. So our second speaker is Professor Koita, who is speaking on COVID-19 pandemic surveillance and network and monitoring, of population of SARS-CoV-2 in the network of diagnosis laboratories in Mali. Prof. Koita.
I would like to thank ICA partner for giving me the opportunity to come. He worked with Don Krogstad, my mentor, who passed away 2 years ago. And they were working in two parts. Part of it was on vaccine development, and Don Krogstad was on the drug development, so targeting malaria. So I will talk today about COVID-19 and to paint, to show the importance of having surveillance program. Just a slide to show the toll of this COVID-19 compared to other pandemics, beginning in 2000s. So MERS is about 850 deaths, and for COVID till February 28, 2023, almost 7 million of deaths. So we should be well prepared for the next epidemic or pandemic. I will not spend a lot of time describing this wording from WHO, considering China. And Mali was in the epidemic in March 2020. And we got several deaths also, like other countries. Here are SARS-CoV infection and also the MERS. As you can see in this slide, sorry. Okay. I see now. Okay.
Can somebody help from the back, please?
Okay. I just want to see to show you the severe pneumonia caused by SARS-CoV-2. You see all by the pathogens. If you have a coronavirus, human coronavirus, just the upper part of the lung is infected. So this may be the reason why we have a severe pneumonia. It's like maybe the virus is a recombine between this one and also between MERS-CoV, because you see the phenotype is quietly having a phenotype from human coronavirus and the phenotype from the MERS. And that's why it was very complicated to protect. Is very important in our country. So the facility we have where we process the specimen from our study site is we was using 8,000 from Beckman Coulter, a gift from NIH, and also all those reagents to set up the COVID lab. And also we participate to Africa COVID, which was a consortium of countries participating to the surveillance of SARS-CoV-2 in Africa. We had other collaboration with some of my colleagues. These slides to show that when we analyze all the specimen using the RT-PCR, we found that the CT, and the CT, when he's low, is a good indication about the burden of the virus. Here we found a CT around 10. And that prompted us to sequence this specific SARS-CoV-2 because we think it may be linked to some mutation, it may be a variant. That was our hypothesis because we could not sequence all the specimens. We just focused on this one because of a very short CT value. And then we followed the patient for about 3 weeks, which is normally had symptoms for about 3 weeks. And we performed a clinical exam, an RT-PCR and a CCT. At the clearance time was May 16. Because the spike gene so when we align all our sequence that we deposit Nigeria and also from Italy, and the sequence was deposited on the gene bank in April. That we knew in Mali that maybe specimen was collecting during the third week with our single specimen. And the third wave, conclusion of this study. Before that, and then we try also to see if efficacious on the SARS-CoV. We did an analysis in terms of change in epitopes. So the vaccine still can be efficacious against this strain. That's why we're more relaxed. And you see there is a big binding between. This variant was very close to the Eta variant, and he led the third wave in Mali. The second, it was highly contagious because of lower CT and highly transmissible. And a huge number of patients is to look at the CT value. If the CT value is less than 24, that's why every time there is a positive, we should know the CT value. And that was a good end. And the second surveillance site we had is the gold mine. It was a partnership between us and the Goldman company in Mali, because they were very careful, do not stop their activities because of COVID-19. So they put money. They asked us to build a P2 lab in 4 sites in Mali, and the strategic site also in Mali, you will see. Exploiting gold in Mali. So the first site that we had was close to Guinea, and is along the Sankarani River, on the Niger River, is at the border with Cote d'Ivoire and Burkina. Third site. This third site was Lulu site, which is at the border of Senegal, along the Falemi River. And those site was very central, so a kind of control for our surveillance. So 4 sites that we monitor for 2 years. So we conceived this lab for the gold mine company and put the same equipment in the 4 sites. That's the second site, the third site. So all the 4 sites, we try to put all the data to manage all the diagnosis of COVID. So there was a partnership between the Minister of Higher Education and the Minister of Health. And that's why the response to epidemic should be across several departments. So in the university, we were able to bring our knowledge to make sure that the data are properly collected, analyzed, and sent to the database at the UNRSP for the Ministry of Health. So then we participate to this consortium, Africa COVID ID, led by the Charity University in Berlin. And the test was based on identify Omicron using this marker, and also include the 2 markers, Delta and Omicron. And then we based on all the Omicron in September 2021. You remember that Omicron was described in November 2021. So in September, only Delta was circulating, no Omicron. And as you can see, the first case of Omicron was in December, normal. In Lulu, in June, you can see no Delta, but Omicron was there in June 21 before November. So 3 months before the appearance of Omicron in South Africa. And that's a point to show that the importance of a surveillance. And in September, nothing in Komana, and in Bamako also nothing. Donc we show that in June, Omicron was present in Mali, it was circulating. And the last slide to show, that's a scale of the continent, all Africa. As you can see, those Delta was present in, July, August 21. And Delta, that will show you how we should be organized in network in order to make sure that we can capture earlier any circulation of a pathogen with a epidemic potential. Very important. And I will conclude that surveillance is a major health activity which is useful, detecting the circulation of pathogen with epidemic potential, and increase of the network of epidemics. And then to think people, all those our colleagues who manage those sites, DJ Ndan for SMK, because there was a physician on site from the company, people from Somilo, people from Morila, ESA, and my colleague Aminata Maiga from Ponji. And also NIH, Bob Watts, was very critical to put the lab in place. Tom Willems, your colleague, Dick Sakai, Cliff, and Sophia Siddiqui. I will not forget for putting money to make sure we put all those data together. And
thank you very much,
thank
bro.
you.
We appreciate you. Thank you. Yeah.
Thank you very much for your talk. I was very interested in the last slide about the spread of Omicron in Mali in 2021. So that paper in Sciences...
Not because of the data from typing Omicron and Delta, but the fact that it was a problem with the sequences that was in the article. But the data that I present are solid. There is nothing. We tried 4 times, same thing. So that the data I presented here is not the reason why it was redacted. It was for the sequencing data, and we did not do sequencing. We just did PCR.
Thank you, Prof. Koita. I just had a quick question. With the CT, the patient that you followed, I don't think you talked much about the clinical presentation for that particular patient.
Yeah.
And because we've had instances where people have CT of maybe, there was a case, one of our colleagues had CT of 11, and her friend who had just arrived had CT of, I think, 18, and she was positive for well over a month, but fully symptomatic.
Yeah, we did not put all the clinical data, but it's in the article that was published. So the symptom was not severe, like the person who need oxygen, no. So that time we say that maybe it's transmissible, but not severe. So we put that clinical data on, it was not severe, it was confined at home.
Okay.
The meaning that it doesn't need any oxygen for breathing. So it was not severe pneumonia.
Thank you.
Right, do we have a question online, please? Yes, there's a question online.
Claudia, could you please ask your question?
Sorry, it was a mistake. I have no question.
I think it was a mistake, so yes.
There's one more in the audience. We can have that.
Thank you so much for your sites. Are you tracking or monitoring the source only focusing on SARS-CoV-2?
One of the slides, it was a SARS-CoV-2. It was not Delta, neither Omicron. They are tracking. So far, it's not Omicron, it's not Delta. Bamako are not Delta, not Omicron. The third column sometimes is 1, 2. It was not Delta, it was not Omicron. Yes, the other SARS-CoV are circulating, and mostly SARS-CoV from base.
Right, thank you very much, Prof.
Thank you.
If you have further questions, you can grab him over lunch or over during coffee break, and then you can have a deliberation in the person of Dr. Diarra, who will talk about circulating emerging viruses in Mali.
Beaucoup. Moi, je vais faire mon intervention en français. Ok, permettez-moi de vous présenter les résultats du territoire malien. Donc, d'abord, je fais une revue générale sur les 3 virus. Le virus Zika a été découvert dans la forêt de Zika, qui a même porté le nom de la maladie, dans les années 1947. Et c'est un virus qui a été classé parmi les flavivirus, généralement transmis par les Aedes, mais aussi par la transfusion sanguine, la voie sexuelle et la voie materno-fœtale. Généralement, l'infection par ce virus est asymptomatique à 80%. Mais on peut observer chez les patients infectés une fièvre céphalée, des conjonctivites et des douleurs rétro-orbitales. Les quelques rares complications donnent l'intérêt à cette maladie, notamment les troubles neurologiques, le syndrome de Guillain-Barré et aussi l'enfant qui naît avec un faible poids de naissance et surtout une microcéphalie. Quant au virus chikungunya, et le virus a été décrit aussi dans les années 1952 au Tanzanie, c'est un virus qui est classé parmi les alphavirus et qui est transmis par les moustiques, la piqûre des moustiques Aedes. Contrairement au Zika, l'infection par le virus chikungunya est symptomatique à plus de 72%, où on peut observer effectivement des fièvres, des céphalées. Mais l'importance aussi de cette pathologie porte sur le fait que ça donne des douleurs articulaires persistantes, plus de 3 mois, pouvant même aller à 12 mois. Et surtout le syndrome de post-chic, où les malades sont souvent déprimés parce que ces mobilités sont limitées. Très proches du chikungunya sur le plan génétique structurel, avec une ressemblance de plus de 85%. Le problème, c'est surtout son diagnostic, parce que le Mali est un pays endémique paludisme, où le paludisme pose un problème de santé et principalement orienté vers cette maladie, le VIH et la tuberculose, ces grandes pathologies endémiques. Alors, il y a eu d'énormes progrès dans la lutte contre le paludisme qui ont contribué énormément à baisser l'incidence du paludisme au Mali. Mais nous avons constaté que malgré la baisse de l'incidence et de la mortalité, c'est pourquoi nous avons décidé d'investiguer les autres causes du paludisme, les autres causes de la fièvre non paludisme au Mali, notamment les causes liées à des pathologies virales émergentes. C'est ainsi, pour d'abord établir un programme de formation et de transfert de technologies en fonction des résultats de surprévalence, mettre sur place les sites sentinelles et de contrôle de ces pathologies virales. Malien. D'abord, en mines différentes du Mali, surtout dans les parties sud différentes. C'était sur un total de 793 échantillons de sérum qu'on a pu récolter sur ces études transversales pour couvrir la partie sec aride du Mali, qui, en 2016, on n'avait pas pu faire à cause de l'insécurité qui règne. On a d'abord testé tous ces sérums par l'ELISA IgG anti-Zika, soumis à des tests de séroneutralisation des virus que j'ai nommés VNT. La participation des femmes était majoritaire dans notre étude. Globalement, dans la partie sud du pays, la prévalence du Zika pouvait atteindre jusqu'à 20% dans certaines localités et 15% aussi bien qu'au sud qu'au centre. Cette prévalence, heureusement, elle est faible dans la partie nord du pays où le climat est chaud et sec. 1%. Alors, en utilisant la modélisation mathématique, nous avons pu mettre en place 2 modes de transmission du virus Zika au Mali, une transmission endémique dans les zones savanniennes où où théoriquement 18% de la population serait infectée dans les années 1995 suite à une épidémie qui est survenue dans les localités. Quant au contexte du chikungunya et de Onyonyong, des virus génétiquement très proches, les tests de sérologie portent toujours des insuffisances pour une meilleure estimation de leur surprévalence. Et comme vous le savez, à cause de la ressemblance structurelle, il y a toujours des réactions croisées entre les 2 virus qui induisent des anticorps et qui se croisent. Donc, sur cette base, il y avait la théorie de l'asymétrie. Alors, donc, asymétrie, tout échantillon était considéré comme positif si le titre d'anticorps est supérieur à 40. Le titre d'anticorps de séroneutralisation. Et si ce titre est supérieur à 4 fois le titre de Onyonyong, il était considéré comme positif. Le sérum est considéré comme oignon positif si le titre est supérieur à 40, mais aussi 2 fois supérieur au titre de chikungunya. Dans les autres cas, le sérum était classé comme équivocal parce qu'on ne parvenait pas à faire le départage entre chic positif et oignon positif. Et nous avons testé sur cette base de séroneutralisation les 1640 sérums du Mali. Nous voyons que ici la partie grise correspond à des sérums qui ont été classés comme équivocales, soit à peu près 10% des sérums que nous avons testés. Ce qui montre que l'ancienne classification porte des imperfections. Alors, nous avons pris, pour corriger ces imperfections, nous avons pris 62 sérums qui venaient de la Martinique et de la Guadeloupe et qui étaient uniquement chic positifs. Et que nous avons appliqué la méthode de classification ancienne. Nous avons trouvé que sur ces 2 îles, il y avait à peu près 16% des sérums qui étaient oignon positifs, ce qui n'est pas vrai parce que oignon n'a jamais été décrit dans ces 2 villes, parce que le vecteur de transmission du virus oignon, qui est l'anophèle, n'a pas été décrit dans ces 2 villes. Ça conclut qu'effectivement, l'ancienne méthode d'asymétrie porte toujours des imperfections. C'est ainsi que nous avons établi une collaboration avec l'unité de modélisation mathématique de l'Institut Pasteur de Paris pour mettre en place un modèle pouvant mieux détecter l'infection à chikungunya et l'infection à oignon, pour qu'en fait l'imperfection qui était dans les équivoques peuvent être améliorées. Alors, une fois mis sur place le modèle, nous l'avons testé par simulation avec 4 scénarios. Premier scénario, le chic est positif à 100%, donc 100% des sérums positifs à chic, 0% des sérums positifs à oignon. Notre nouvelle méthode mathématique détecte exactement 0% de oignon et 100% de chic, comparativement à la modèle classique où le chic revenait à peu près à 31%, contrairement à 100%, et 22% de oignon, contrairement à 0%. Alors nous avons fait 4 scénarios, et ces scénarios nous ont permis que notre modèle détecte mieux les chic et oignon que la méthode classique. Ainsi, sur la base de de ce modèle mathématique, nous avons effectivement trouvé que la prévalence du chic au Mali peut atteindre 20% dans certaines localités et que encore toujours au nord, dans les zones sèches, semi-arides, cette prévalence était faible, environ 3,7%. Globalement, la prévalence du chikungunya au Mali, en tenant compte des 1640 échantillons, était de 13,3%, et que la prévalence de oignon au Mali était environ de 30%. Ça, c'est normal parce qu'il y a les vecteurs qui circulent sur tout le territoire malien. Pour conclure, nous avons, à partir de ces 2 études transversales conduites sur tous les territoires maliens, nous avons détecté la surprévalence, la circulation du virus Zika à 12%, chikungunya à 13% et oignon à peu près 30%. Grâce à la modélisation mathématique, nous avons pu décrire des modes de transmission du virus Zika au Mali, endémique dans les zones savaniennes et épidémique dans les zones semi-arides. Les faibles taux de Zika, de chikungunya et oignon ont été observés dans la partie nord chaude où le climat est chaud et sec. Je vous remercie.
Merci beaucoup. Right, so to the audience, questions for Dr. Diarra, sorry. Questions from the audience? Yes, one hand.
Thank you, Prof. I have a question. I don't know if maybe you have the answer to this, but looking at, I think mortality from chikungunya is quite high, at least the official mortality rate. So I'm wondering if you have such a high seroprevalence. Is this something that translates?
Okay.
And then the second one is related. Zika, sometimes, I think usually people are worried about the second infection with Zika. Désolé. Okay. I'll start with Zika.
Okay.
I'll start with Zika. Zika, usually you worry about the second time you get Zika because it's supposed to be that time that you can get serious complications. So with such a high seroprevalence, is this something that is correlated with a spike in hospitalizations for Zika? And the first one was about chikungunya. Is it also correlated with deaths from febrile illness?
Ok. Merci beaucoup. Si vous regardez un peu l'introduction, je peux dire avec réserve que c'est la première étude où on a détecté d'abord la surprévalence des virus, les 3 au Mali. Mais il y avait d'autres études qui donnaient des surprévalences à partir des échantillons de sérum des banques de sang. Les cas cliniques, à mon avis, à ce que j'ai entendu jusqu'à aujourd'hui, il n'y a pas eu un cas clinique de Zika qui a été décrit, il n'y a pas eu un cas clinique de chikungunya, il n'y a pas eu un cas clinique de nyonyok. Tout simplement parce qu'ils ont les mêmes symptomatologies que le paludisme et que le paludisme était considéré comme un problème majeur. La prise en charge de tous les cas de fièvre non diagnostiqués était soumis à un traitement antipaludique, un antalgique et un traitement antibactérien. Ce qui fait que le paludisme et que peut-être ils vont donner un intérêt médical qu'on doit investiguer en deuxième phase, mettre des études cliniques sur ces sites sentinelles de haute prévalence. Donc, je pense que j'ai répondu aux 2 questions en même temps. Merci. Oui. Ça, c'est une question culturelle. C'est les femmes qui s'occupent de l'unité. Quand on appelle la population, c'est les femmes qui se sentent toujours contre. La participation volontaire des femmes, elle vaut 2 fois que celle des hommes. Donc, moi, je pense que c'est tout à fait culturel et comportemental.
Moi, je voulais savoir s'il y avait des conditions afin de pouvoir concevoir éventuellement des systèmes d'alerte précoce qui permettraient de capter épidémiques. Qu'est-ce que vous pensez de ça?
Effectivement, la deuxième phase de cette collaboration, parce que dans la plupart des cas, c'est pour désormais investiguer des géographies, pour nous aider à faire la géoposition, tout ce qui est explication facteurs climatiques. Mais déjà avec la modélisation, vous avez vu que la transmission elle est endémique, savanienne, semi-aride et sec chaud. Donc il s'agit avec une équipe multidisciplinaire de mettre cliniques et voir leur co-infection avec le paludisme, quelle peut être la sévérité ou la cause de mortalité. Donc, on en prévoit. On demande.
And thank you, audience. At this juncture, I'll call on our next. So the question online.
There's a question online.
Sorry. So, Dr. Diarra.
France. Yeah. So thank you very much. The question is from Francine. She said, thank you very much for this nice presentation. You said that ONNV, onion virus, was not circulating in one study site because the vector Anopheles is not present. Do you not think that there may be secondary vectors acting there? Another question is, as I know, PRNT is a gold standard technique for serology. Is there no way of differentiating between a chick and onion infection? Additionally, why didn't you target also dengue virus? Thank you.
Alors, je m'excuse de prendre les questions en retard, mais est-ce que vous pouvez les reprendre s'il vous plaît? Ah, c'est gros.
Okay, so come back to the first question. You said that onion virus was circulating in one study site because the vector is not present. Do you not think that there may be secondary vectors acting there? First question. So the second one is, I know that PRNT is a gold standard technique for serology. Is there no way of differentiating between a chick and onion infection? So finally, why didn't you target also dengue virus?
Oui, en se basant sur les tests sérologiques classiques, comme vous l'avez dit, PNRD, et classifier les sérums à 100% chic positif ou 100% onion positif. Il y avait toujours un nombre de sérums dont vous parlez, qui est en 2015. Il y a une épidémie de chikungunya sur ces sites. Parce qu'il n'y avait pas d'anophèle, c'est les îles françaises, il n'y avait pas d'anophèle qui avait été décrit. Donc, tous les positifs à la modélisation mathématique. La deuxième question, quand j'ai nommé notre test de stérilisation de virus VNT, Virus Neutralization Test, vous le savez, le PNT, c'est un test classique ancien. Et c'est très délicat parce que vous utilisez du gel à chaud, vous risquez quand même d'avoir des soucis. En plaque de 96, on parvient à tester plus de 88 échantillons. C'est rapide et plus besoin de faire recours au gel. La lecture se fait tout simplement en regardant les effets cytotoxiques des virus vivants sur un tapis cellulaire, comme vous le savez. Donc, avec mon collègue, et on a publié le papier en 2020. Donc, c'est la nouvelle, c'est l'alternative de PRNT qui est VNT. Et le titre de positivité, c'est que le titre soit supérieur à 40%. L'avantage alpha-virus, les autres flavivirus sont négatifs. On a testé avec des gens vaccinés qui a vraiment fait la différence entre Zika positif, fièvre jaune positif, dengue positif. Donc c'est pourquoi nous l'avons utilisé comme nouvelle technique de vaccination.
The person of Dr. Podono. Thank you.
My name is and get us started. Our first session talk is by Dr. Fermin Kra from the University of Alassana Ouattara.
Okay. Good morning. My name is, initially I plan to speak in English, but how always go to speak in French on behalf of the Français. Alors, J'ai fait cette communication à la demande d'un groupe d'auteurs. Donc, au lieu de faire la communication en visioconférence, nous avons fait le choix de faire une communication en présentielle à travers ma voix. La question qui va nous occuper ici est de savoir préparer à prendre en compte les dimensions sociales des questions des épidémies ou de notre esprit. L'importance ou l'enjeu de le cas de la récente épidémie en Afrique de l'Ouest, épidémie de la maladie à virus Ebola, et aussi les épidémies de la peste successives de la maladie à virus Ebola au Congo. Et plusieurs acteurs en sciences, plusieurs auteurs répondent efficacement à ces épidémies sans prendre en compte les questions sociales, culturelles et épidémiques. Et les analyses de nos expériences sociales ont montré qu'il faut évidemment adapter les mesures sanitaires au contexte culturel et politique. Nous avons deux réseaux en sciences sociales se sont mis ensemble pour sociaux, de sorte à pouvoir intervenir sur les questions sociales en contexte d'épidémie. Et nous avons le réseau anthropologie des épidémies émergentes, non, le RAE, qui a été créé en 2014 durant l'épidémie de la maladie à virus Ebola. Et il a pour objectif donc de partager les expériences entre les chercheurs, comparé d'Ebola qui était financé par Expertise France dans 5 pays d'Afrique de l'Ouest. Ensuite, nous avons le réseau SONA global qui est financé par l'Union européenne à travers son projet H de 2020 et qui est coordonné par l'Institut Pasteur à Paris. C'est un consortium de 18 institutions, et donc, dans le réseau anthropologie des épidémies et le CSRF. Et l'objectif de SONA Global est de créer une masse de scientifiques sociales, de sorte également à partager les expériences, les méthodes et les outils sociaux. Et donc, ces deux réseaux se sont donc mis ensemble pour mettre en place des modules de formation pour le renforcement de capacités. Donc, en 2017, en novembre 2017, les deux réseaux ont mis ensemble les dispositifs, ce qui a permis entre 2017 et 2018 d'également destiné à la formation 250 personnels de la santé. Et sur 5 ans, ensuite, en 2019, avec les deux l'anthropologie des épidémies émergentes, on l'a fait également. Voilà, et ça nous a permis d'obtenir ces résultats. Donc, on a produit 22 vers la fin de l'année 2022 qui contient. Et vous pouvez les consulter sur le site des deux réseaux qu'on a créés. Donc, les modules que nous avons faits ont permis donc d'obtenir 13 chapitres, donc 13 modules de formation dont les contenus sont là. Ils abordent globalement les questions politiques, les questions culturelles et aussi la question du contexte d'émergence des épidémies, donc le contexte social, les mesures sanitaires et les questions des rituels funéraires lors de ces épidémies. Donc, en conclusion, il faut dire que pendant ces années, et en 2022, au Burkina Faso, et aussi plusieurs formations, suite de la formation continue toujours. Et nous sommes toujours aussi en collaboration. La question, c'est que c'est au niveau des pays pour que nous allons travailler ensemble pour que les pays, les autres pays puissent se. 3. Nous envisageons une formation de 25 chercheurs en sciences. Je vous remercie.
Excellente présentation. Constraints.
Thank you very much in this continent to link social sciences together with infectious disease sciences. Do you have any concept in how to bring across such training modules, not only to social sciences, to anthropologists, but to people like those here in this room, beyond?
S'il vous plaît, la translation.
Can't speak any French. I try. Also addressing people like here in the audience who are more working from the biology, from medical sciences, to link, end up linking social sciences with this kind of research which is described here. Did you understand?
Yes.
Ok. La question est de savoir s'il existe des modules qui sont adaptés à des gens comme dans cette audience, c'est-à-dire des biologistes ou des chercheurs, etc., parce que ça serait très important et très intéressant. Donc, est-ce que vous faites des modules, pas forcément pour les gens qui sont en sciences sociales déjà, mais également pour les biologistes et les chercheurs? Merci.
Merci.
Donc, si je comprends, la question, c'est la formation des biologistes. En gros, oui, je pense que dans le programme, il est... Mais aussi, on parle aussi des scientifiques sociaux, avec des formations pour les biologistes, s'approprier les approches anthropologiques. Des clous qui
on emerging health challenges and building surveillance systems within the One Health framework. I would like to invite Dr. Oliver Comey, Dr. Janet George Lorient, Dr. Benedicta Mensah, and online we have Professor Folarin from Redeemer University in Nigeria. So if you all want to come up. So welcome to this session. I'm going to just pass the mic to my co-presenters here to give a short introduction on themselves and their work.
Good afternoon. My name is Joseph Oliver Kome. I work with the Ghana Health Service. I'm currently the acting director for the Ghana Infectious Disease Center. I'm an infectious disease specialist. I'm happy to be here.
Hi, my name is Benedicta Mensah. I'm a research fellow at the Noguchi Memorial Institute for Medical Research, and I am interested in looking at transmission dynamics of infectious diseases. Thank you.
Good afternoon. My name is Janet George Lorient from Sokoine University of Agriculture, Tanzania. I'm an assistant lecturer, but also a postdoctoral research fellow for SACI's African Center of Excellence for Infectious Diseases of Human and Animal. I was one of the first beneficiaries of AC2 in Tanzania and East Africa. So I'm so much privileged to be here and share my experience. I'm a One Health economist and systems analyst.
Thank you. Do we have Professor Folarin?
Yes, good afternoon, everyone. I'm greeting everyone there in Ghana from Nigeria. So I am Professor Onikepe Folarin. I am the Deputy Center Leader for African Center of Excellence for Genomics of Infectious Diseases, and we are based at Redeemer University. Nice to have you all.
Well, thank you, and thanks to our panelists. So the COVID-19 pandemic has reminded us of the inequalities of existing health systems across many African countries. Although significant progress has been made in building health surveillance systems, major systemic deficiencies still remain. No one can deny the close links between human health, animal health, and the state of the environment. Therefore, there's a need for greater collaboration between health research institutions across national boundaries to optimize the use of the available capacity to benefit all of us. So this symposium's first roundtable, current and past pandemics, and how to build better health surveillance systems in Africa, and most of all, to promote my attention to Dr. Kome. Dr. Kome, can you tell us a little bit about what the Ghana CDC is doing in building innovative and efficient surveillance systems of transmittable disease? And can you also highlight some of the challenges that you're facing?
So thank you very much. I'm sure we were invited to this table. Diseases were done in silos. But then with the advent of COVID, we were privileged to have a center, an infectious disease center built to respond to future pandemics and future infectious diseases. So that's how I'm moving towards trying to have a semblance of the Nigerian CDC. So we have the Ghana CDC. Also, we think that we should respond to future pandemics attached to it. And we believe that the lab is supposed to more or less serve the surveillance aspect of it. We are part of the Ghana Health Service Network, so we are open to everybody. Before COVID passed, move things forward. And I think for where I sit, you highlighted, yes, the difficulties with research. For my institution, we can only be effective if we collaborate with the academic institutions. Going forward, we know that some of these infectious diseases are there. Currently, we are dealing with what we say is an outbreak of Lassa. I'm sure Lassa has been here for a long time, but we just haven't looked for it. Now we have an infectious disease center. We are managing the Lassa cases. We are collaborating with research institutions. It's not just about making diagnosis. But recently we had Marburg, and we were told that have we done. So I believe that it's important to make diagnosis, but it's important to understand or know the virus. And we can look at how the future epidemics, and know that maybe, let's say in the next 2 years, prepare to have maybe Lassa. We need to position ourselves well and learn how to collaborate. Yeah, that agenda forward. Is it just the ID people just talking about it? How do we collaborate with microbiologists? How do you collaborate with our friends who are experts in genomics to understand? And in future, even come up with vaccines, candidate vaccines, to manage some of these infectious diseases. So for me, I believe that the advent of COVID, we should not limit ourselves just to the clinics. Let's talk about using CT values to just predict the people you need to. So I think if we have, I believe sometimes it's for infectious diseases, the laboratory capacity and know how they are behaving and how to manage it. For me, sequencing is the way to go. It's just not RT-PCR and we are there. Understand the disease, understand the pathogen, know the history of that pathogen, and then we can move forward.
Collaboration, multidisciplinary collaboration, and genomic, francophone, anglophone countries, sharing of knowledge, and how do we work as one team. So with that said, can you tell us a little bit about
area of project researchers and partners working in in strategies, in promoting strategies to enable us respond to the COVID-19 pandemic. And one of the main objectives was we achieved this objective by a collaboration of IRD, Noguchi Memorial Institute for Medical Research, and WACCBIP. We conducted 3 repeated independent, the COVID-19 prevalence, seroprevalence, 8 months period that we conducted positive, being seropositive compared to the younger, 3 times likely the time.
Well, thank you. It's your results are a bit similar. What's interesting is I'm in the so again, this is a scenario where we have to tell. I'm glad to see that there is capacity building. Dr. L'Oreal, and how can we leverage your work to better improve surveillance systems?
Thank you. Maybe also wanted to have this beautiful One Health surveillance system, because Tanzania is so wanted to move to that direction of having One Health systems, because to do the surveillance. The roadblock that I encountered the very first time was that we have a stronger public health system, and our animal health surveillance system was not that much strong surveillance system. And then I started right now we have, and then also we had our digital, our innovation, the digital surveillance tool called Afya Data. So Afya Data is collecting data from the community and using syndromic approach, while the MI, which is FAO innovation used case-based events. And then we also have laboratory information system. So thinking, is there a way we can have this integrated system? Because once we manage to make it interoperable, it's easier to see maybe in one dashboard what's going on in the laboratory, what's going on in the field as people they are doing their normal operations, and the three systems, all they reflect what we call the API, the data, the formula that we are using to pull data from laboratory to see that at least now we can minimize the threshold. If the threshold to announce that we are able to detect the disease just on the onset, because if you see cases, the disease is endemic, but then the threshold value has increased, it's also an alert for us to follow up. But also we can have laboratory confirmation that this is a positive case and it's notifiable. So that way, with that kind of integration, to me, I was comfortable that now with the animal health surveillance as integrated as it is, can be an input to the One Health digital, to the One Health surveillance system. And then the next step now was we got a fund with our colleagues from Ending Pandemics to develop now the One Health platform, where now we are integrating the system from animal and human and all other early warning systems at country level, so we are able to respond as a One Health.
That's fantastic. Issues. Yeah, that's. It would be amazing if what you've done can also be replicated in other countries. What will you think we need to do to be able to scale what you've done? I mean, it's really amazing.
I'll go back to Dr. what he said, collaboration. We are trying to engage with the FAO. For instance, we have some countries that pledging to work with us, especially on integration of the countries that have implemented the MI and our innovation, the AFIA data, to integrate. So we are packaging this integration as a package, so that we start from the community. Those who are able to start from the community, they can do so, and then we have this official reporting line. But that can only be possible once we collaborate, because you can't, you may know what's our share experience from other countries through collaboration and it may be less expensive compared if you want to hire a consultant to do that.
That's a very excellent point. Again, it comes back to collaboration and partnerships. Thank you. Professor Folarin, are you online?
Nigeria.
So, all diseases in West Africa. Can you please share with us your experience and how do we optimize regional?
I want to appreciate the organizers of this program and for inviting me, even though I'm supposed to be in Ghana physically with you, but due to some other engagements, I'm not able to be there. But in terms of our experiences concerning surveillance of infectious diseases, we at SGID, we confirmed the first case of the disease, and then we also, CAT actually brought us to start thinking that it's not going to take any special effort for any other part, preparing for any other future human capacity through series of genomics next generation sequencing training that we also train bioinformatics analysis so that they can be available for genomic sequence genomics. The second thing that we have also done is to develop infrastructural capacity to give us an edge for high throughput system for extraction and library preparation. And we also invested in the different next generation sequencing platform, such as the Illumina and as well as the Oxford NovaSeq 6000 center in the country for the detection of COVID-19. And we were also designated as the reference lab for detection of pathogens within the country and also across the region. So with respect to our experience on COVID-19, we actually sequenced the first case of the virus in the country within 48 hours of receipt of the samples, and this was, and so immediately we equally sequenced over 7,000 samples, and we have also assembled over 5,000 genomes of the SARS-CoV-2, all of which are presently available on GSAID. We use the sequence data to defect of the virus, and this is basically what we do for most of any outbreaks that we have. Then one thing that we also, particularly the Presidential Task Force on COVID-19, aside of sequencing samples from Nigeria, I'm talking about COVID-19 now, we also sequenced the many other countries. And we also did similar thing in different countries where the samples came. So going back to collaboration. So I would say, and they would help us to coordinate the surveillance process. This way, expertise will be identified and then knowledge will be shared across the region. SGID, in collaboration with Africa CDC, WHO, we conducted training on next generation sequencing for the detection. We trained scientists from Cameroon, Cote d'Ivoire, Seychelles, Rwanda. Overall, we covered about 30 for training. We also went to the different countries to help them to establish their genomic laboratories. And we also trained them on site for COVID-19 sequencing. So this is what we need in Africa. We have been able to tell our own story, especially with COVID-19. Thank you.
Professor, that's very impressive work in Ghana from the infectious disease standpoint. I think it would be great for all the ways to meet and for this kind of collaborations to happen. I mean, it's amazing work that's being done, and I'm really impressed. And I just want to make sure that this doesn't end here, that we all come here and have a nice meeting, and it all ends up here. So again, congratulations for such amazing work. Dr. Mensah, tell us a little bit about what Noguchi is also doing. I know you're doing some great work, and we want to highlight what you're doing and how you can also contribute to the sub-region pandemic surveillance program.
Similar to promoting the One Health approach. So we have ongoing initiatives and programs, and one of them is the Emerging Infectious Disease Program, which focuses on identifying and response to new and re-emerging infectious diseases. So we conduct research on emerging infectious diseases such as COVID-19, Ebola, and Lassa fever, as he said. We provide capacity through next generation sequencing and also specialized diagnosis for Ghana Health Service, and we also support the Ministry of Health. And we actually house the national, we are the reference center for polio nationally, and then regionally we are the center for influenza, if I haven't switched that up. And then we are also, we also have the One Health surveillance approach, where we focus on a comprehensive surveillance system, as she also described, where we integrate human, animal, and then also environmental data in detecting and also response to outbreaks in a timely manner. We are also interested in looking at antimicrobial resistance as well. We want to also develop strategies to combat anti-malaria resistance and also develop new antimicrobial agents. And then we are, finally, we are also interested in vaccine development. We've been involved with vaccines towards Ebola, vaccines like COVID-19 and malaria.
question goes. What this organization stands for, WANIDA stands for. I will not be speaking for Ghana, because then if I speak only for Ghana, I think that my scope will be limited. But being here and seeing the capacity in other countries, I will go back to my question of saying, let us collaborate. We will sit down, come up with a plan. We cannot focus everything in one country. So let's say Senegal is going to handle for us maybe vaccines, Ghana handles for us diagnostics, Nigeria handles for us NGS sequencing. Then we have the rich experience from Mali telling us clinical things. I'm sure if you ask what we want to do to benefit everybody rather than just one small country. Now is collaboration is going to be key to solving our problems here and come up with moonshots. Moonshot for this very French Development Bank for helping put this symposium.
Another round of applause for our panelists. Okay, it's time for lunch, but before we go, a few housekeeping items. You may have noticed in the program that we have a networking lunch with HCA. Going on at this very moment, we have a workshop by the Human Cell Atlas group. And the idea is that we will share the lunches so that there's collaboration between, there's conversation between the groups that are sharing this time and space at WACCBIP. And then maybe some ideas will come out of some of those conversations. Some of our faculty and students are involved in that, and some are involved in this. But we couldn't find any other way to sort of merge the two events. So hopefully this lunch will be that. And lunch will be served outside in the car park. When you exit just to the right. There's also posters which are on the first floor in the main biochemistry building. You've moved them? So for the Americans, second floor. For the British and Ghanaians, first floor. So I think that's it. And we'll meet back here at, what time? 2:30. Thank you. Bon appétit.
ATI or don't, that's a good discussion to have. It sounds like it would fit in with some of the things that you are interested in. So what exactly is your work that you're doing out there? Okay, great. Yeah, that's interesting. That's something that's so dramatic and so sudden that it comes in, starts spreading in communities, and it really is a unique opportunity to describe those things for COVID. So one thing, so what we're doing right now, the way that we're doing, showing that this vaccine works, is vaccinate, have a control group, randomized study, control, vaccinated. And then every way we do it now, we have to learn best how to do it, but every 2 weeks we put the mosquitoes on people's forearms and let the mosquitoes feed for 15 minutes and second the mosquitoes a week later. And so we, not a lot of those are positive, probably like 1 to 2% of those screens. These are not taking a blood smear and seeing if they're positive blood smear. It's just vaccinated population. A lot of people happen to be carrying parasites, and so you'll get a measure of how frequently people are transmitting when you put mosquitoes, 60 mosquitoes on them. So that, takes out some of the factors of attractiveness and things like that. It's really, is this a person that's transmitting? Because the mosquitoes will feed since they're starved. And then we get measures of, was there a transmission event? If there was a transmission event, what proportion of the mosquitoes got infected? And then one thing we haven't looked at is the burden. We have the measures, but we haven't tried to examine it. We've solely done our vaccine trials, analyzing to what, our efficacy, so to speak, is how have we reduced the number of positive feeds? How have we reduced the number of positive mosquitoes? And then there's that dimension about also osis burden, which has been, we're sort of interested in sporozoites also. But there's a real complexity there that it's interesting to imagine how you can take those data and restructure them to predict what's going on in the community. Now we have had some interface with Tom Churcher and Joe Challenger from Imperial College. They have their malaria models. They have models. They had a paper in Nature Communications that said if you combine RTSS with the transmission blocking vaccine, this is the effect that we would expect to see. So there's been some work on that, but there's just a lot more that could be done, and especially. Okay. Okay.
With this trial, you establish, and we see that the human being infected with
similar tools to start to get those some metrics about how frequently those events are happening. Which what part of the population is most likely to transmit. There's we have these practical issues about how we can most efficiently measure in a vaccine trial. We need, the problem we've had is getting enough events, surprisingly. Despite the fact malaria transmits, so explosively in Mali, it's pretty impressive when you feed mosquitoes on people that it's a very small fraction, 1 to 2% of the feeds that are positive, blood smear. So we have those data as well. And, a large majority of those who transmit do have a positive blood smear, but there are cases that they don't have a positive blood smear. Clearly they have parasites and they transmit. And then when the gametes come out of the red cells in the midgut, that's when the antibodies can attack them when they come out. It's really fascinating. That started in the 1970s, the concept of the transmission of antibodies. Yeah, shall we walk over there? You can leave things to be said, right? Oh, thank you. Back.
Est-ce que on a la télécommande de ça juste les 30 secondes?
Should I put it up here?
Yes. Okay. Perfect. Thank you.
My name is Joseph Olilakome. I'm the acting director for the Ghana Infectious.
Could you tell us how was table going on? Was it good? Was it interesting? What did you talk about?
Okay, so I took part in the plenary on how we can maximize surveillance to look at infectious diseases. I must say that I was impressed with what I heard from my colleague from East Africa and also from Nigeria, and also the work that is being done in Noguchi and Wade. I believe that the key word here for me is we need to collaborate. If you remain as a one center, you are not able to achieve a lot. But then when you collaborate with other centers from different countries, what you think is a difficulty for you, somebody might be doing it very easy. And what that person thinks is a difficulty, you may. So we can harness our efforts together. And I'm sure we can have a solution to an African solution to the infectious diseases that really worry us more than any other place.
Okay. I'm just thinking if I have another question. Why is it important for you to be part of this WANIDA symposium?
Okay, I must say that maybe I stumbled into this symposium by accident. I happened to meet Olivia as an excellence or ACES of ID is key. For my center, we are just 2 years old. So for me, coming here, maybe we will say it's an accident, but I'm happy to be here and seeing what others are doing elsewhere gives me great motivation that if we work hard, we shall also get there.
Okay, that's okay for me. Thank you very much.
Thank you too.
Thank you.
Thank you.
Tous les moyens en anglais que tu as aussi été très étrange. Un collègue à qui parlait là, qui c'était sur une tétanisée. C'était un peu une habit. The idea is to really have a regional approach. What's the name of the pandemic? Pandemic first worldwide.
never know what the level is. That's all that up to a senior person to influence this person. If it has a heavy teaching load, there needs to be a real 9% voice like that. Is advocating for that and trying to establish shared policy.
I'll see you later.
I guess I could have looked around.
Hello, 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 everyone. Call to order, please. Right, so welcome back from lunch, everyone, and we continue with our next session for today. Our next theme is looking at strategies for vector-borne disease control and elimination. For this section, we have our chair in the person of Dr. Peter Kwashi, who will chair this session and then call the presenters to be on stage. Dr. Peter Kwashi, the podium. Right, thank you.
All right, good afternoon. Welcome back to the afternoon edition. So without much ado, I'm going to call, oh wait. Sorry, I'm a little lost. Okay, yeah. I'm going to call Dr. Claude Ovry, one of our keynote speakers, and he is the head of Global Health Development Program for Malaria at Merck, and he's based out of Switzerland. And he's going to present on insights on Merck Global Health Integrated Innovation and Partnership Strategies as a way to transfer decisioning to African stakeholders.
Okay, that's a long title. I'm not sure the presentation gonna reflect exactly the title. I'm here as a duo because I'm inviting my colleague Delali, and he's based in Accra. Okay. And he's the representative of the Merck Global Health Institute in Accra, and for all, and he's running all our African projects here. Okay. So he'll talk about the projects himself. So before starting on, maybe if we can start the slide so we don't lose too much time. And you can actually move to the next slide, but before dipping into the presentation, what I need to say is we are actually representing Merck Germany and not Merck Sharp and Dohme. So there is two different pharma companies in the world that are calling themselves Merck. So one in Germany, one in US, and the one in US is the one doing vaccine. We not. And so that's just for the landscape. Now Merck in Germany has actually three different business. One is the healthcare, where we develop medication, so specialty on oncology, immunology, immuno-oncology. And there is a global health department within that, and I'll come to that. Second business, I'm sure you will be familiar with, is what we call the life science business, and it's anything that has to go to supply and consumable for lab, but up to building factories for production. And if I give you names such as Millipore, Sigma, you will know what I'm talking about. So this belongs to Merck's portfolio. And the third business is about, it's called electronics, and it's the modern 21st century chemistry. And we're moving really quickly towards the interaction between the nanotechnologies and the cell biologies and that type of thing. So here the key products I would say for public would be the liquid crystal that was managed and produced by Merck, as well as now the LED and the OLED technologies. This is technologies that has emerged by the company. So with that, actually, I will quickly go through the global health intent and where the Merck Global Health Institute fits in. Then Delali will show you the activity that we have in Ghana and that we want to expand across the continent. I mean, we wish to, not we want, but, and then dipping into a little bit more how we manage the development of a new drug for malaria, which has quite nice promises. So starting with global health at Merck, so next slide. I can do, sorry, let me be, yeah, so again, I mean, the advantage to have these three businesses is that you can go across the portfolio and the expertise and the know-how of the three completely different business, all related to life science. And you can build, you can take out the technologies, the assets that are there and develop them for specific needs that underserved population will have in low and middle income countries. This is what the intent of the Global Health Unit is about. And within that unit, the research and development part is managed by the Merck Global Health Institute, where we both work at. And so the global health strategy, we have three key pillars. The first one is of course driven by, is driving being a key driver of the elimination of schistosomiasis. This is driven by the fact that we are giving 250 million tablets per year to WHO, of praziquantel, sorry, to WHO. And around that Lighthouse program, we have developed research and development activities for diagnostics, higher sensitivity diagnostic, moving away from catocats to RDTs. We also just in a process to have registration by EMA of a pediatric formulation of praziquantel. And all that is to increase and accelerate the impact of the use of praziquantel. The second one is tackling other global health challenge. Here today, we are mainly focusing on malaria, and this is what the talk will be today. And there is a third group that is more looking at innovative ways to get access to low and middle country of the portfolio of Merck. So how can we change get better pricing for these countries? So again, at the Merck Global Health Institute, we are doing the research and development. We have, again, we're looking for technologies, existing and emerging technologies that we can transform and adapt to the need we have. We pretend to be science and data-driven. We pretend to do science, and I think we do science. And what we want is to enhance the output of our data by using artificial intelligence, data science, and digital technologies. The only way we can do that for us is through partnership. Merck is not an infectious disease company, so in order to advance the portfolio, the only way we can do is to do our research in partnership. So it's other internal partners, but most of it goes outside and it's looking for external partners. And there is a talk that follows this one, and that will talk about a research program that it's a technology about identifying, characterizing drug resistance for new anti-malarial drugs. Technology that we transfer from the expert lab David Fidock in New York, and we implemented that in Mali, same lab as David was talking this morning. And not only we transfer the technology, but we transfer the question that we had to the lab. And they were they had the responsibility to build up the answers and to come up with that. Of course, it was a group work, but this is the way we want to develop technologies and build capacities in Africa. We want the brain, not so much the technique. And this is an outlook of some of the activities we have and collaboration that we have across the continent. Just as a note, doesn't look like a lot. We are 20 and about 5 researchers within these 20 persons. So just as a note. And now I hand over to Delali for.
All right. Thank you, Claude. So my name is Delali. I'm based in Accra, Ghana. So I handle all the projects that we run in Ghana. And we tend to pilot a lot of these projects from Ghana and intend to spread it out onto the continent. So like Claude was saying, as Merck as an organization, we tend to tackle malaria as one. That's why we call our malaria initiative as one against malaria. And for this initiative, we have different projects or different pillars that we integrate together to try and eliminate and combat malaria on the continent. I'll be speaking on 3 of these, which are currently running and piloted in Ghana, and Claude will hit on one that he is the real lead on. So the first project I would like to just briefly give you some insights on is what we call the Pan-African VIVAX and Ovali Network, which we call PAVON for short. So the vision of PAVON is to have an Africa free of Plasmodium vivax and Ovali. Yes, the fight is always on Plasmodium falciparum, but studies have shown that vivax and Ovali have begun to emerge in Africa. Our mission is the mission of PAVON, is to drive the elimination in Africa through epidemiological survey of PV and PO. The main objective is to establish a network, support data-driven policy making. And not just that, we also train the next generation of scientists for Africa. So I'll give you some background on why we started this PAVON network with our partners. So in Botswana and Namibia and Zambia, we carried out a study to study the ecology of PVVAX infection in these countries. So basically, these countries have very low incidences of or prevalence of falciparum, but we went on to study if there is a presence of PVVAX and PVVAX in these countries. The results clearly identified the presence of PVVAX in Namibia and Botswana in the northern parts of these countries, and also determined that these PV patients were definitely antigen negative. What this has done has caused policy change and also treatment and diagnosis change in these countries. The PAVON network has also gone on to train some scientists in these countries and have been approached by the Ministry of Health in these countries to be the official partner for surveillance network in these countries. So out of this PAVON network, we have 3 PhDs thesis written to. So this just gives you an indication of where the presence of the PVVAX is in the country. So the red spots on the right will show you the prevalence rates and where they are, the hotspots where they are concentrated. So all these areas we thought were malaria-free, but the survey or the study has shown the emergence of PVVAX in this country, in these regions, sorry. I said earlier that most of our operations start from Accra or Ghana and expand. So as part of that, we have established what we call the PAVON Research and Training Facility here in Ghana. This training facility is just to build the capacity of African scientists in malaria, in the malaria space. So at this center, we have it's completely off-grid. It's on solar. It has dedicated high-speed internet, and we are in the process of establishing and equipping molecular and biological laboratories in these facilities. So we are basically in the process of doing that. So we don't do just the surveillance, we do also capacity building and training for the next generation of scientists. I'll briefly also talk on another project we are running in Ghana called the IR3535 project. Here, we are doing a local study to evaluate the repellent efficacy of this molecule IR3535. So IR3535 is not a new compound. It's been around for about 40 years, but we are trying to repurpose it to see if it can be effective against the Anopheles mosquito and also contribute to the fight against malaria. So basically, it's chemically stable and can be used for different formulations. So together with a partner in the US, they've done a new formulation which is much, much longer acting that we are testing in the field and in the laboratory to see if it will be effective against the Anopheles mosquito. So we do this together with partners. We do this together with partners in Ghana and overseas to establish this efficacy. We do this through a 3-phase approach. We do a laboratory test to check its efficacy against the Anopheles mosquito, which we have successfully done. Now we are currently running the field study to see its effectiveness in the natural setting. So the last project that we are currently running in Ghana is called GALAC. It's a capacity building program for medical lab scientists in the public sector. So basically, we train together with partners, the Noguchi Medical Institute and the Ghana Health Service. We bring them in into the Noguchi Center, once a year to train them on malaria microscopy and other comorbidities that come with malaria. We have done 2 pilots and have successfully trained 50 participants, but the next step is to expand into other parts of Africa. So we are looking at expanding into Nigeria, Kenya, then we move on to other continents that are of need, such as the South America region. So these are the projects we're running in Ghana. I leave it to Claude to continue. Thank you.
Thank you. Wake up. So yeah, the next part is to explain you a little bit what happened if you're all doing research, you're all having ideas about, like David this morning, identify potential drug vaccine candidates. What come next? So here it's about a molecule that was initially identified by the University of Dundee and collaboration with MMV, Medicine for Malaria Venture. It's the first EF2 plasmodium inhibitor. It was published in 2015 in Nature under the short name of DDD498. So that was the code name so Merck in-licensed it, so it's inhibiting this, the elongator factor 2 with a high sensitivity compared to the human target. This is the graph on the left, the right, over the show. And so Merck has in-licensed this molecule in 2015. Key characteristic of DDD498, which became M5717 when it becomes to Merck, and is now named cabamyquin, which is the WHO official name. So I will use cabamyquin in the presentation. Will make things a bit easier. So the molecule is active against all dividing stages of the parasite. It's EF2 is an enzyme that is involved in the translocation of mRNA into protein, and so the molecule blocks the protein synthesis. One thing we really have discovered recently is that by blocking the parasite protein synthesis, you actually change the sequestration phenotype because there is no expression on the surface of the red cells. So that's something that is discovered. We need to talk. So we in-licensed that protein at the beginning of the animal testing stage. So what's the regulatory, the regulation says is that you need to test, prior to go to a human trial, you need to have 2 species tested, one rodent and one non-rodent. So here we choose the rat and the dog. And the problem is the molecule has a very long half-life. It's over 150 hours. And the normal design of such experience is that you dose daily the drug. And the objective of the preclinical stage is to identify what are the target of the organ of toxicity so that when you transfer it to human, you know what to follow, you know where the potential risk will be, where you will see adverse event. The problem there is, since the drug is long, if you dose every day, you're going to have accumulation of the drug, and this is exactly what happened, and it was fully toxic, and we couldn't distinguish anything. So we had to go back, think a little bit about a different design, and so we looked at the pharmacokinetic of the drug in the 2 species, and we look at the time point where the dose of the drug was close to 0. And then with that, we came up with a protocol where we would dose the animals for 3 days, and they giving a space with no drugs and come up with the next dosing. And with that, we successfully identified the target organs, and we could define also the first human human safe starting dose. So the minimum dose you can use in human in your first in-human trial, your human volunteer study. And this is what we did. So then after the preclinical study, you go to a phase 1, so human volunteer, where you do initially a single ascending dose. So you give 1 dose, and you slowly increase the amount of drug up to the point where you start seeing adverse event. So for us, we started to see adverse event in 1800 milligram. We still went up to 2.1 gram. And we stopped here dosing after two patients because we had confirmation of the adverse and we didn't want to expose more. We did that trial in Australia, in Brisbane, in James McCarthy clinical unit, and James has developed the human blood stage challenge model. So it's a model where human volunteers will be, Australian students for most of them, will be receiving blood stage parasite, malaria parasites, and when they reach a certain level of parasitemia, they have the experimental drug giving. So that allows you to have an early readout of the activity of the drug on the blood stage. So there we tested 400 milligram, 150 milligram, 400 and 800 milligram. So, okay. So in terms of safety, what we've seen is that the molecule is pretty safe. We could raise the drug up to 1800 milligrams, so nearly 2 grams of drug. And most of the events we've seen here were mild, transient, and with no sequelae. So pretty safe drug. And when we looked at the efficacy, actually, which is the interesting part here, on the right you have the 800 milligram where all the volunteer infected were actually totally cured. There were no parasites remaining. On 150 and 400 milligrams, we had recurrent parasites. So we had 3 in 150 milligram that had parasite that came up, and 2 in the 400 milligram. When we looked at, we sent this recurrent parasite to David Fedock in New York for sequencing. Actually, 2 out of 3 and 2 out of 2 parasites in the 400 milligram were harboring genetic mutation that were associated with resistance in vitro. So that means that the parasite has a very high propensity to resistance. So you give the drug once, bang, you have resistant parasite. That's pretty big red flag for the molecule. But with that, we still decided to continue because we think that we can mitigate that with combination partner. So that's very similar profile as for Atovaquone, for example. The next study we wanted to do, remember, the drug is active against the liver stage, so we wanted to also demonstrate that the molecule was active into the liver. To do that, the key question here was, what is the dose that we can test, okay? And in order to do that, we had the modeling and simulation the model that we derived from the human volunteer, the phase 1. We also had experiment in mouse where we could define an IC50 in the mouse. And the other thing we did was we have developed a 3D hepatic cells culture that we can infect with sporozoite and develop cells culture, hepatic liver infection cells. And that we use that as well to do the testing of the drug and the activity. And with that, we modeled the efficacious dose, and what came out of the model was that a dose between 60 and 200 milligram would provide between 30 and 100% protection. So in order to protect the patient, we decided to move with the 200 milligram dose, and in the sporozoite challenge. Here, what we did was to really look at, mimic the chemopreventive drug. So we infected the volunteers with the cryopreserved sporozoites, wait for 2 hours or 5 days, and then treat the drug. And the result here, the Kaplan-Meier curve, shows you that the 100 and 200 milligram gave 100% protection, while with already 30 milligram, you start to have an activity compared to the placebo. So if we go back to the previous slide here, that means that the 3D cells we've been developing here actually is pretty predictive of what you're going to see as a dose, allows to do a pretty good prediction about what you're going to see in the human models. And that's an advancement, because with that we can also reduce the use of animals, and that was also an interesting aspect. So here again, we've shown that we confirmed the activity in human of the liver stage and the blood stage. And if you then came the clinical development, so moving towards patients, and this is where we are today. So if you think about treatment for malaria today, there is the WHO defined three different use cases of drugs. So the case management, treating patients, prevention, which is basically avoiding patients living in malaria endemic country to become sick. And then there's a more tricky place. The third one is on the elimination, which is more on mass drug administration, etc. So having blood stage and liver stage, we thought that we could actually develop this drug for at least the first two indications, and this is what we're starting to do now. But again, one of the things that WHO is recommending is that each and every new anti-malaria has to be developed in combination, and also knowing that we have a high propensity of resistance to carbamazepine, we actually went to the quest of the partner drug. And it took about 2 years to look, and it's work that we did in collaboration with MMV, we did collaboration with Novartis, etc. So we're really looking across each and every available molecule, and the criteria we had were really of selection were really selecting a drug that would protect carbamazepine against resistance. So first thing is you needed to have a matching PK, so the exposure, the duration of the presence of the drug in the blood have to match, have to be at the same length of time, so that the two drugs will protect each other. They needed to target a different enzyme or site of action, mode of action. And the drug itself should be having a low propensity to have resistance itself. So, end of the game, we choose pyrrolizine. So pyrrolizine is actually developed in combination with artesunate under Pyramax. So there is a lot of safety and efficacy data around that molecule, which allows also a certain acceleration and de-risking of the program. So that's another positive asset. So we are now on the point to dose the first patients. We have two phase 2A trials that are ongoing. One is to cure the patients, so having classic case management, 28 days protection. The other one, we are dosing asymptomatic patients, and with that, we will have a big data-driven decision point on how we're going to go further with this drug, either for cure, either for prevention, or being more clever and doing the two at the same time. So it's a long way forward still. I hope we can accelerate things, and but let's bet for success. And today I would define success as, let's hope that one day both the malaria and the Women's Day will be out of the calendar. That would mean a lot of SDG achieved. Thank you.
Thank you, Claude. So we have time for a few questions. In the back there.
Okay, thank you for this nice talk. In the beginning of your presentation, you mentioned that the molecule can also block sexual parasite during the transmission from human to mosquitoes. And in the development, you don't have an idea about how the specific target on the parasite on which this drug molecule can act on parasites. Can I have an idea?
Sorry, you have to articulate and speak a bit louder.
Okay. My question is mainly on the effect, transmission blocking action of the molecule on the sexual parasite. At the beginning, you mentioned that the molecule can block parasite transmission. I would like to know the specific target on the parasite on which the molecule can act.
So the official identified target is the protein elongation factor 2. So that's an enzyme that is involved in transformation of mRNA into protein. So it's actually, the action is blocking protein synthesis. So I see its gametocytes are not dividing stage, but there is still an effect on it.
So before fertilization, you mean?
Yep.
Okay.
So all that is in the blood, but if you go back to the paper that was published by the group in Dundee,
okay,
you will see that you have activities even in the mosquitoes, etc. So it's really pan-specific. It acts at the same nanomolar concentration, so it's equipotent as opposed to a lot of other antimalarial where you have like 10-fold difference between the liver, the blood, and the gametocyte. So we have additional data on that we've generated on gametocytes, so it's confirmed. And part of the phase 2 that we're running in Burkina and Gambia will do DMFA as well. So we'll have the direct effect after treatment on the patient.
Okay, thank you.
Can you look in vitro or in parasites for development of resistance?
Sorry?
Can you look for development of resistance to the molecule?
We did that, and I think that's the topic of the next call. I mean, we did it a lot because, of course, I mean, it's a red flag since the beginning. And of course, we know that. We did a lot of characterization with David Fiddock, and so he's in New York. And one of the things that was interesting and triggered the next, there is a huge fitness cost to the resistance. So basically the parasites are outgrown by non-resistant parasites. And so the question we had is, what's going to happen in the field? And so what we did is to
Claude, could you clarify one point? I don't think I quite understood. There were resistant parasites that came out in the phase 1 study with James McCarthy.
Yep.
So I'm not a drug person. That seems extraordinarily rapid for resistant parasites to come out of a controlled infection. So what are your thoughts? Clearly you're trying to get a partner drug, and I understand, but just how does that happen so quickly?
So one hypothesis is, I mean, it's not a hypothesis, it's work. That's part of the drug we did with David, and we confirmed with Laurent in Mali, is that it's apparently pre-existing resistance in NF54 and 3D7. I mean, the thing is, each and every, from in vitro up to human volunteer, you're using laboratory strain, and it's the same strain. Where what we want is what happened in the field, and actually it's quite different. And I don't know, the explanation and stuff. It's clear that it's a flag, and this is something we will follow, because even prior to investing to next phase 2B, where you go to 50 to 100 million investment, then it's something you want to understand. So it's known in bacteriology that EF2, so when they do antibiotic development, EF2 is considered as a non-druggable target because of resistance. So in bacteriology, they just forget the target. Right.
I have two questions. Well, a comment and a question. First, I think I'll be interested in visiting the Pavon
with
site.
pleasure. It's there for that. And the other thing I didn't know, but I didn't say, we do a lot of modeling and simulation for drug development, but also for epidemiology, etc. this is something we want to actually bring somewhere, and probably in Ghana.
Okay. And then the second question was, in your clinical trials, when you did your dose escalation, and you got to, I think, 1,800, and with no toxicity.
Mm-hmm.
This was despite the fact, were you dosing every day?
It's single dose, actually.
Oh, okay. Okay.
Yeah. So it's one dose. Normally what you do after single dose is you go to what they call the multiple ascending dose. So it's the same, but you give 3 or 4 dosing.
Right.
But here the intent is really to give that drug as single dose because of the half-life. So we decided that we had sufficient exposure for what we needed to do.
Okay, that makes sense. Okay, thank you very much.
Thanks to you. And by the way, very pleased and honored to be here. Thanks, Olivia.
So now we have there's 2 online questions. Claude. And they will precede 2 online presentations. So the first online from Francine. Thank you for your nice presentation. I wish to know if Merck are organizing. Yes.
No, we don't. So again, it's data driven, and we can do if it's associated with one of our research projects. And basically, what we do with funding, and there's 2 examples in Pavon where we had like 3 trainer to master student that were part of Pavon, and that will presenting this at the conference.
Mm-hmm.
The work that was presented as the inception of Pavon was 2 PhD students as well, financed as research project. So that's the way we do it. We don't have like free grants, at least not in our department, possible at Merck, but I don't think so.
Okay. And the second question is how decision making is shared between Merck and the partners in Africa and Europe.
It's so we're a very strong believer that it has to be shared. It's consented decision. It's again, we're looking for brain, not so much for technology. Of course, technology is important. What we want is, the uniqueness of the partners. So you know the field, you know the conditions, you know this. So for us, it's really, I don't know, I'm Swiss, so, it's the difference between the Swiss and the French government. So it's either centralized and vertical or organized. Mali is really that. We transfer the question to him. Okay. And of course, it's working together. And I really believe that if you want to have strong decision, hierarchy needs to go away. In any sense, for the decision, when you look at the science, and then the implementation has to be structured. Okay. But
all right, thanks
again. Philosophy here.
And so now we switch gears a little bit. We have 2 online presentations, and the first will be by a colleague of Claude's, Claudia De Mata, who is a global health scientist at Merck, Germany, and she'll be presenting the value of P-falciparum field isolates for the prioritization of drug combinations to de-risk malaria drug development in the case of, I forgot, carbenequine M5717. Okay.
Hello, can you hear me?
Yes, we can hear you.
Okay, I will share my screen. Share, and wait. Okay, can you all see my screen? It's fine, to be sure.
Not yet.
I'm sharing the presentation.
Yes, it says you have started screen sharing.
It's fine?
Not yet.
Okay. Look. While we are waiting, so good afternoon to everyone. So I'm that researcher that Claude just mentioned, and I'm working at Merck in Switzerland with him, and also I'm one of the researchers that is working with the different laboratory in Africa regarding malaria and schistosomiosis program that we have.
Okay, we see your screen now.
Perfect. So as Claude just mentioned, I'm going to present the combination and how we studied the combination with carbamazepine and pironaridine with our collaborator, Professor Dembele in Mali. And this was based everything. We had a question. We wanted to do it with field isolate. And also we wanted to implement an assay that was developed in Europe with 37 and NF54 laboratory strain, and wanted to see if it was possible to have it in Africa using field isolate. So coming back a little bit, so as Claude just mentioned, carbamazepine is a novel anti-malaria drug, and and also is an elongation factor 2 inhibitor, so no protein synthesis, and is addressing the unmet need for malaria treatment, especially for the most vulnerable population, such as children and pregnant women, because the compound was tested safe in the different toxicology study for these two populations. So what we did and what we observed during the carbamazepine development, using laboratory strains, so 3D7 and NF54, we did a minimum inoculum for resistance study, where different inoculum of parasite were exposed to the drug, to carbamazepine, and after some time, and we monitored the killing of the parasite and also the recrudescence of the parasite after some days. And what we observed with carbamazepine is that 60 days after the exposure with the drug, we have some recrudescent parasite. What we did, we took this parasite, recrudescent parasite, and we did some genetical analysis to see what was different from the original parasite, or if we still Y type or something happened. And what we highlighted here in the graph here is that we highlighted that we had different mutations with different degrees of resistance. So the red one are the higher resistant one, and the green and the low resistant parasite. And at this point, because we had all this data in vitro, we wanted to know if what's happened in vivo. Can we have also the same development of this resistance in vivo? And if yes, how we can mitigate this resistance, maybe finding the new a right partner. So here what we did first, we used the humanized mice model, so the energy mice model, engrafted with human red blood cell and Plasmodium falciparum parasite 3D7. We treat the animal when the parasitemia in the mice was around 1 with carbamazepine. And what we observe a killing of the parasite, no parasite in the blood, but 20 days after we treated the mice, we saw that all the mice treated presented recrudescent parasite. For some of them, what we did, we retreat them with carbamazepine, and what we observed in 2 out of 5, that the parasite was completely resistant to carbamazepine. Indeed, we didn't see any killing of the parasite or control of the parasite. And for 3 of them, retreated with carbamazepine, we saw a partial response to the treatment. For all of them, we took the recruiting parasite and we did as before the genetical analysis. And what we observed, it was that the recruiting parasite are mutant parasite and resistant to carbamazepine. And now at this step, we decided, okay, as we know, we need to have a partner drug, as is also the mandate of WHO that we cannot have anti-malaria treatment with only one single drug. So what we need to find? We need to find a partner drug that have a different mode of action of our compound, that has a similar half-life of our compound, because if one of the two, if we have a mismatch in the half-life and one of the two will persist as a monotherapy, we can develop some resistance to the second compound. And also, if it's possible to use a drug that is already in the market that is known to be irresistible. For this reason, as Claude mentioned before, we choose to work with pironaridine, that is a well-known compound. Also because pironaridine is a fast-acting drug, inhibitory of beta-hematine formation, and even more specifically, pironardine is matching our half-life of carbamazepine, that is around 150 hours. And also, there are no resistance reported for pironardine at the moment. So we redid the humanized mice model study where we infected the mice with Plasmodium falciparum, and we treated them with the combination of carbamazepine and pironardine. And what we observe is that we have a delay of recrudescence parasite after day 20, so they recruited around day 40. Only 2 out of 5 have present recruiting parasite. We retreated them with carbamazepine alone, and we were able to completely kill the parasite. And at the end, we have all the 5 mice that were completely cured. So at this point, to expand the limit data set that we obtained from mice while limited the animal use, we developed a new in vitro interaction parasite reduction ratio assay for early de-risk the clinical development of anti-malaria combination. So briefly, this was developed last year. It was published last year too. So briefly, in this new assay, what we do, the concept here is it was to mix the 2 compounds at different concentration and check the parasite viability at different time after drug exposure using parasite reduction ratio. And take all these viability data and using in silico modelization, try to fit all the data and obtain the different parameters that allow us to have some idea about the drug-drug interaction, and also to check if our dose prediction in human was correct. However, this assay was developed with laboratory strain, so with this assay, we could not really take in consideration all the genetic diversity that we can have on field. And for this reason, we decided to implement using field setting for the fields. We decided to make it accessible to field setting research and obtain more data vis-a-vis the parasite genetic difference in collaboration with Professor Dembele in Mali. So basically what we did here, we adjust the in vivo study assay we did before. So we adjusted, so taking freshly field isolate from patient, and we started an ex vivo culture for 72 hours, because it's the maximum time that we can culture field isolate parasite in a mix of our 2 compound concentration, and we measure the viability of the parasite, and with this viability of the parasite, we were able to modeling the rate of killing of the 2 parasite using the combination, and see where we had limit, and also if pironardine was a good partner drug for carbamazepine. And the next step, it will be to use the parameter obtained from this model to always in silico simulate the dose that we can use in clinical trials. So basically, the first step that we needed to change in this assay to allow it to be used in a field setting, it was that we developed the first assay using parasite reduction ratio step, because if we do a normal cybergreen or microscope analysis, the problem is that we can only have information about what is detectable. So we have the information about parasite growth and that we can detect. But we have up the limit of quantification. But all the parasites that are under the limit of quantification, that are invisible with the cybergreen and sometimes also at the microscope, we cannot detect them, and they are really important for the analysis. For this reason, and since Professor Dembele worked on this micro-tracker assay, we assess the use of cybergreen combined with a micro-tracker readout to try to identify the viability of the parasite, as is shown here. Viable parasites were in life, so green and blue. and that parasites are characterized by the blue light. And this allows us to continue our study. So basically, once we have all these, we had obtained all these viability data, what we did, we put together a grow parasite model in silico. This model is a one compartment model that is summarizing the killing and the growth of the parasite. When the parasite is in monotherapy, is under the monotherapy or combination therapy. And with this model, we were able to obtain the different parameters that we didn't obtain and we cannot obtain using a mice model. So the first step was to analyze the monotherapy. Here is the monotherapy of carbamazepine. So what you can see is that the parasite growth was well described by the model. So the dots are the raw data obtained in the laboratory, and the line that you can see is the line obtained by our mathematical model. So as you can see, for carbamazepine, we observe a regrowth, a low dose. You can see it more at 1.8 nanomolar or 7.5 is more clear. So we see a initial killing of the parasite followed by a regrowth of the parasite. This is suggesting that the parasite are developing an adaptive resistance to the drug, a low concentration, but not a high, because at high concentration we have the complete killing of the parasite. So using this model, we were able to derive the EC50 of carbamazepine that was around 20 nanomolar after we have this full adaptive resistant development, so when we have high dose of our compound. For pironaridine, everything was fine. As before, the model described very well the data. And here we didn't see any adaptive resistant development. And we were able to model that the EC50 of pironaridine, that is a constant IC50 and is around 30 nanomolar. Now, we wanted to study what's happened when we combine pironaridine with carbamazepine. So first, we check, as before, the viability of the parasite. Using them, we observe that when we give carbamazepine plus pironaridine at the same dose where carbamazepine was, we observed this just adaptive resistance, pironaridine was capable to completely kill the parasite, also the resistant parasite to carbamazepine. So after what we did, we took all the data, the parameters that we extrapolate from the model, and we did extrapolation, and we did a heat map to try to visualize the killing rate of the parasite. And what we observe is that if you check here, so at 30, we have the EC50 of pironaridine, and 6 nanomolar, we have the EC50 of carbamazepine. And when you observe, when we exceed both drug EC50, we have a completely kill, and the killing rate of the parasite is stay constant. So meaning that we will kill all the parasite without having this adaptive resistance that is coming out. Only for you to know, when is blue-green, here is meaning that the killing rate is really fast, but when is pinky, we have a slow killing rate. So now, as Claude mentioned, before, and also I just mentioned, the problem are the resistant parasites. So what we did, we redid this MIR study done at the beginning using laboratory strain in Africa. So we implemented the assay in Professor Dembele lab. And we tried to obtain some resistant parasite to carbamazepine field isolate. It was a really hard work because it was really hard to obtain resistant field isolate to carbamazepine, but we succeed. So we obtain one resistant parasite, and we use it into this combination assay and see what's happened when we use this resistant parasite. And what we observed that it was really nice is that it seemed that the two compounds have a synergistic interaction. Indeed, as you can see here, carbamazepine, when we put the carbamazepine into the parasite culture, we have no control of the parasite growth, of course, because the parasite is resistant to carbamazepine. And the same things is happening when we use subtherapeutic dose, so low dose concentration of pyronaridine. For example, 5, as you can see, the parasites still regrow without control. But what's happened when we put them together is that the combination of the drug...
1 minute.
It's finished. The combination of the drug is able to control the parasite growth and completely killing the parasite. So it's meaning that carbamazepine potential the killing effect or pyronaridine. So in conclusion, we were able to develop this assay for field isolate, and also able to develop a model that can mimic the field parasite growth. We saw carbamazepine also with field isolate displaying an adaptive resistance, but that with pyronaridine we can control this adaptive resistance and we can have a stable killing rate all along our treatment. So I want to thank my colleague at Merck and also our collaborators, so Dr. Dembele and his student in Mali, and Dr. Sebastian Vika that helped us for all the modelization part. And thank you.
We can take a few questions, and
Yeah.
I'd like to kick it off. I'm a little still going back to the earlier talk. I'm a little surprised at having such a long half-life and the quick evolution of resistance. Do you know if the resistance mutants affect the half-life of the drug at all?
No.
This is when we're talking about mono.
No, the resistance, they are not affecting the half-life of the drug. The half-life is still the same.
So does that mean that, so that's you're measuring the half-life of it bound to the EF2, right?
Mm-hmm.
So does that mean that this can progress even in the presence of the drug bound?
Sorry, I didn't understand your question.
In the case of a resistant mutant,
Mm-hmm.
where you have resistance, but then you
Mm-hmm.
have the drug present. I'm just trying to picture what that looks like.
So if you have a resistant parasite and you put the drug
So
is
it's not a
is
competitive inhibitor, is what I'm asking. Is it a competitive inhibitor or is it?
No.
Okay, I think I'll ask Claude because maybe I'm not asking it properly. There's a question.
Thank you very much. My question goes thus. The principle behind the use of artemisinin-based combination treatment is because we want to have two drugs, one short half-life that kills large biomass. Your carbamazepine with propensity of resistance. And then we are trying to mitigate that propensity for the parasite to develop resistance with pyronaridine, which is currently in use. And this is still at the experimental stage. And we know that one of the factors that drive resistance is drug load. Now, using pyronaridine currently, which is becoming popular, and then these are experimental stage, what happens if we have tendency of resistance by the time this passes through all the phases? Then what is the plan that we have to circumvent the parasite being adapted to the drug at that level before we begin to look for this alternative? Thank you.
Do you want to answer? Oh, I answer. Oh, I.
That's a very good question. And that's also a reason why we want to have two drugs having the same duration, because one of the weakness of ACTs, which we see happening, is because you have resistance against ACTs, you start to have resistance against the long-acting drug, because there is no protection, because it's short half-life, and you have like pyronaridine is for 40 days without protection, so it's a single drug use actually. ACTs is 2 times single drug if you want. Here we try to have two drugs that are fast-acting, so killing the parasite a little bit together, and then protecting the reemergence together. So that's a little bit next generation of combination if you want. It doesn't stop your question about if there is resistance of a pre-existing resistance of pyronaridine. It's true, and that's a risk. Pyronaridine, no one has ever been able to generate resistance. So it seems that it's a drug that has a very, very low propensity to have a resistance generation, which we never know. If, as you said, if there is suddenly an expansion of use, that may happen. We expect that by combining, you can overcome the existing, but yes, it's a risk. Then we'll have to switch to another combination. But to be honest, I mean, there is no such new drugs existing, actually. If you look at, the pipeline, and if you look at the portfolio of MMV, we went through all of them. But technically, that is now ready to go into phase 1. There are 2 or 3 drugs. So, and if you take out the one that have short half-life, and there was one drug we were discussing with Novartis, but which is really an artesunate-like, and I don't remember the name, but the code is KE609. It's super fast-acting. They're testing it into severe malaria now. But the fact is, the half-life is super short. And so when we put them together in mice, bang, you had resistance to the 2 drugs at the same time. So it's not so much choice, unfortunately. But don't ask us, ask MMV.
Okay.
Is combination as hybrid molecule rather than a combined drug. What I mean is that currently we have hybrid molecule where you have 2 anti-malaria with different mechanism of action that we chemically combine them together. So it's like having 2 different drugs as one. And the idea is, can we deceive the parasite by having a different molecule which combine 2 different molecules with different mechanism of action? So that is what I'm just thinking, that is it possible at this stage to be looking towards that direction? Thank you.
I think it's possible. It has limitation as well. One of them would be, first of all, you need to have the same ratio of drug if you want to, because then, if you need like 2.5 more drugs of one versus the other one, and then it's certainly a cost, and that would mean a new complete development, because it would be a new drug. So yes, but it's a way to look at it. I mean, there are other ways where you could actually link the molecule to different things that actually potentiate the effect. There was, Patrick.
Okay. Can I ask that we hold the rest of the questions till the evening so that we...
Sure.
All right. So we have another online presentation by Dr. Freddie Bryce Simo Nem, and he's a postdoctoral fellow at the Center for Research in Infectious Diseases at Yaoundé, and he'll be presenting the clinical, biochemical, and virological study of the co-circulation of Plasmodium, SARS-CoV-2, dengue, and chikungunya in febrile patients in 2 hospitals in Yaoundé. This sounds like a very interesting talk. Do we have him online? Do we have him online? All right. Dr. Bryce, if you're speaking, we can't hear you.
Allô, je ne sais pas si vous m'écoutez là.
Okay, we can hear you. Do you mind making your slides slideshow?
Bien, bonsoir à tous.
Bonsoir.
Si le monde m'écoute, bon, j'ai un problème avec la webcam de ma caméra, de mon ordinateur, donc j'utilise mon téléphone en même temps. Je ne sais pas si le son est bon. Ok. Merci pour cette opportunité que vous m'avez donnée de pouvoir présenter ce travail qui est intitulé.
Hello?
Oui. Yes, yes.
Can you show slideshow? Slideshow. Yeah. No, the slideshow.
Selection. Ouais, tout.
Tu peux faire le mode diaporama?
Oui, j'ai mis un diaporama sur mon ordinateur. Je ne sais pas si c'est visible. Can see it there. Je pense que c'est pas visible. Une minute, s'il vous plaît.
Okay, let's go on. Let's go on. Commence.
Ok. Bien, merci pour cette opportunité que vous me donnez de pouvoir présenter ce travail intitulé Études cliniques, biochimiques et virologiques de la co-circulation du plasmodium, du SARS-CoV et de la dengue et du chikungunya chez les patients fébriques dans 2 hôpitaux de la ville de Yaoundé et du district de Yaoundé 4. Il vous sera présenté
Est-ce que vous m'entendez?
selon le plan ci-contre.
Est-ce que vous m'entendez?
Oui, oui, je vous entends.
Est-ce que vous m'entendez?
Oui, très bien.
Ok, ce que vous pouvez faire, c'est vous enlevez le partage, vous partagez tout votre écran, ensuite vous repassez en mode présentation et vous partagez l'écran. C'est ça, partagez maintenant tout l'écran. Tout l'écran. Voilà, maintenant, revenez sur votre présentation. Donc là, ce qu'on voit, c'est un dossier avec des fichiers Excel. C'est ce que nous voyons.
Oui, je reviens sur...
Je suis sur la présentation, mais je ne sais pas pourquoi ça n'affiche pas.
Voilà. Maintenant, passez un mode diapo pour voir. Vous avez étendu votre écran ou pas? Vous avez 2 écrans?
Oui, oui, oui. En fait, j'utilise l'écran de mon ordinateur.
Ok, présentez comme ça. Je pense que...
Bon, je ne sais pas, on va se débrouiller comme ça. Donc, le plan de la présentation est le suivant. En guise d'introduction, la fièvre se caractérise par...
S'il vous plaît, augmente le volume.
La fièvre se caractérise par une augmentation de la température au-dessus de 37 degrés en absence d'effort physique. Et c'est l'un des mécanismes innés de défense de l'organisme face à des agressions étrangères. Donc, l'étiologie de la fièvre peut être infectieuse. La plupart du temps, on retrouve des allergies ou non infectieuses causées par des allergies ou infectieuses. Donc, la cause majeure des pathologies fébriles parasitaires en Afrique centrale ou subsaharienne, on trouve généralement l'infection par le plasmodium ou bactérienne par le typhus. D'autres causes mineures, à savoir des infections par des arbovirus, ont récemment été démontrées et des études ont montré la circulation des virus dengue et chikungunya et plus récemment avec la pandémie de la COVID-19, ce virus fait partie des causes de pathologies fébriles. Ces différentes pathologies présentent comme commun une fièvre d'apparition brutale couplée à plusieurs autres signes cliniques dont les plus principales sont des courbatures, des céphalées, de l'arthralgie, des myalgies et puis des douleurs abdominales. Sur le plan immunitaire, du moins, ces pathologies, sur le plan biochimique, ces pathologies se caractérisent par une élévation du taux de créatinine, phosphate, des plaquettes et de cellules lymphocytaires. Dans le monde, ces pathologies se retrouvent généralement dans la zone intertropicale, à savoir des infections par le plasmodium, la dengue et le chikungunya, avec des prévalences allant de 10 à plus de 90% selon la région. Et plus récemment, le COVID-19 a été montré circulant en Afrique dans plus de 47 pays et touchant plus de 3 millions de personnes. Des études antérieures ont montré la co-circulation entre le virus de la COVID-19 et celui de la dengue, notamment rapporté au Brésil, à La Réunion et puis en Colombie. Et des facteurs tels que l'âge, c'est-à-dire des personnes ayant un âge de plus de 65 ans, ou des facteurs de la colocation avec des personnes malades, une dyspnée, c'est-à-dire une température supérieure à 39 degrés, et puis les facteurs immunitaires tels que le taux de créatinine élevé étaient des facteurs qui sont généralement associés à cette co-circulation, co-infection entre ces différents pathogènes. Dans le cas du Cameroun, quelques études de prévalence ont été effectuées et ont montré effectivement la circulation des virus dengue, chikungunya, et le plasmodium est le pathogène le plus circulant dans le pays. Mais des études montrant la co-infection entre ces différents pathogènes restent à ce jour inexistants et peu d'informations sont présents sur les facteurs qui pourraient être corrélés à la co-circulation entre ces différents pathogènes. Ainsi, face à cette observation, nous nous sommes posé la question de savoir quels seraient les facteurs qui sont corrélés à la co-circulation entre ces 4 pathogènes chez les patients fébriles venus en consultation dans les 2 centres hospitaliers du district de Yaoundé 4. L'objectif de ce travail était donc de déterminer ces facteurs corrélés à la co-circulation entre ces 4 pathogènes chez les patients fébriles venus en consultation dans ces 2 centres hospitaliers de Yaoundé 4. Et plus spécifiquement, il s'agissait de déterminer les caractéristiques de ces participants, de déterminer la séroprévalence et la prévalence des co-infections entre ces 4 pathogènes, ainsi que de déterminer les facteurs liés à la co-circulation de ces pathogènes. Pour atteindre ces objectifs, nous avons mené une étude transversale et descriptive à l'hôpital de district de Biémassi et au cabinet médical Saint-Étienne, dans le même district, pendant la durée allant d'octobre 2020 à janvier 2021. Donc, nous avons inclus durant cette période un total de 181 participants présentant une fièvre d'apparition brutale datant de moins de 7 jours avec un ou plusieurs autres symptômes, à savoir la trachée, les céphalées, les douleurs abdominales et bien d'autres. Dans cette étude, nous avons exclu des patients présentant des fièvres chroniques et des patients non consentants. Cette étude a fait l'objet de l'approbation par le Comité national d'éthique de la recherche et les autorisations de recherche du district de santé ont été obtenues. Ainsi, pour les patients consentants, ces derniers ont été soumis à un questionnaire suivi d'un prélèvement sanguin, dont une partie a été utilisée pour la numération formule sanguine, une autre partie pour la recherche du plasmodium par GTS et une dernière partie pour la détermination du taux de créatinine phosphate, des antigènes dengue, des IgM, anti-SARS-CoV et anti-chikungunya. Les données ont été analysées à l'aide du tableur, ont été rentrées à l'aide du tableur Excel et puis analysées à l'aide du logiciel SPSS. Et les facteurs associés à l'infection ont été déterminés à l'aide du test de KID. Suite à cette méthodologie, les résultats suivants ont été obtenus. Le tableau suivant présente les caractéristiques socio-démographiques de la population. Nous notons que la plupart des participants étaient des femmes et c'était âgés de moins de 20 ans qui étaient plus des élèves et n'avaient aucune connaissance et la plupart des participants n'avaient pas de connaissances spécifiques sur les arbovirus. Pour ce qui était la prévalence de ces infections, l'infection par le plasmodium a été détectée chez plus de 70% des participants, suivi de l'infection de la présence d'anticorps anti-chikungunya chez 30% des participants, suivi des IgM COVID chez 14% des participants et enfin de l'antigène NS1 de la dengue chez 1% des participants. Donc, ces prévalences sont proches de ceux obtenus par H1D et collaborateurs en 2016 qui montrent une forte prévalence élevée du plasmodium chez les patients fébriles. Et les résultats de WACCBIP et collaborateurs en 2018 montrent une faible circulation de l'ARN du virus de la dengue chez ces mêmes participants et une circulation du chikungunya chez 30% des participants à cette période. La figure suivante présente les co-infections entre ces différents pathogènes. Nous notons une forte co-infection entre le plasmodium et le virus chikungunya chez 17% des participants, suivi d'une triple co-infection entre le COVID-19, le chikungunya et le plasmodium chez 4% des participants, et seulement 0,5% des participants étaient co-infectés par le virus de la dengue, par le virus de la dengue et celui du COVID. Donc, ces résultats sont les résultats de BAC et collaborateurs en 2020 ont aussi montré une faible co-infection entre le virus de la dengue et celui de la COVID-19 chez 25% des participants dans une étude chez les patients fébriles menée au Brésil. Les tableaux suivants présentent les résultats des corrélations entre la présence des pathogènes et les différentes caractéristiques de la population, du moins des différentes caractéristiques cliniques des participants. Donc, nous avons juste pris ceux qui avaient présenté des corrélations positives, à savoir la présence de douleurs articulaires, de courbatures et de douleurs musculaires a été associée à la présence du virus chikungunya, tandis que la présence de douleurs thoraciques a été significativement associée à la présence du virus de la dengue. Par ailleurs, pour ces paramètres biologiques, la présence de la thrombopénie a été significativement associée aux patients présentant le paludisme et la présence d'une CRP élevée a été associée à la présence de chacune du moins à la séropositivité à chacun de ces pathogènes. En conclusion, donc, la co-circulation du SARS-CoV-2 de la dengue et du chikungunya et celle du paludisme a été effective, a été démontrée dans ces centres hospitaliers de la ville de Yaoundé 4, bien que le paludisme demeure une cause importante de la fièvre au sein de la population. Ces autres infections devraient être prises en compte et chez les personnes hospitalisées, la possibilité de co-infection a été aussi matérialisée, du moins a été la plus élevée. Les paramètres cliniques et biologiques caractérisent spécifiquement la présence de ces infections, comme nous l'avons dit, la présence des douleurs articulaires, et dans le cas du chikungunya, la présence du moins dans le cas du COVID, les douleurs thoraciques. Et ces paramètres devraient être mieux investigués afin de servir d'éléments de prédiction de l'évolution de la pathologie. Merci pour votre attention.
Merci beaucoup. Unfortunately, we don't have much time for any questions because of the technical challenges. So we'll move straight on to the next presentation, which will be by Dr. Etienne Bilgo, who's a senior postdoctoral researcher at the Wellcome Trust Fellow at the Institut de Recherche en Sciences de la Santé, CEA ETEC MTV Burkina Faso, and he's also an ARISE grantee. And I'll let him talk about his topic.
Thank you very much, Peter, for this presentation. So, good afternoon, everyone. So I'm the last speaker. It's going to be an exciting talk. No worries. So my talk is on mosquitoes, mostly on mosquitoes. But you're going to see that there is relationship with mosquitoes and COVID at the end of my talk. So first of all, I want to say thank you so much for WANIDA for giving me such great opportunity to present some data that we just collected from our lab. So my talk is on how to use Wolbachia and fungi to control mosquitoes and like vector disease like Aedes. So a recent paper has shown that maybe in 2018, around 60% of the world population gonna face to vector-borne diseases. That means around 6 billion people in the world. So this is mainly due to a lot of parameters like the growing urbanization, but mostly on the insecticide resistance also. You can see here in this figure, this is fogging of insecticide in Malaysia. But even by fogging a lot of insecticide, they are not able to control Aedes. So that means we need new tools to support the common tools that we have in our hands if we want to do a lot of progress. So we are proposing to use Wolbachia to support our vector control tools to control dengue. What is Wolbachia? Who is Wolbachia? Wolbachia is a gram-negative intracellular bacteria. It's naturally found in around 60% of arthropods in the world. And Wolbachia plays two main characteristics. First of all, Wolbachia is a parasite of mosquito reproduction system. And secondly, Wolbachia looks to confer some protections to insects when they're having Wolbachia in their body. So that means we can use Wolbachia to make refractory mosquitoes to viruses or to make them refractory to malaria parasite, for example. So, and Wolbachia is a parasitic microbe of mosquitoes reproduction system due to this mechanism called cytoplasmic incompatibility. We used to say CI. And this cytoplasmic incompatibility is led by some genes called CIF genes. And for example, I'm gonna make a short caricature of the system. For example, if a male of mosquitoes has Wolbachia and is mating with a female which don't have Wolbachia, which doesn't have Wolbachia, the female cannot lay eggs and cannot contribute to the next generation. That means we can use Wolbachia to suppress mosquito population. And this is the aim of one of the program in the US called Mosquito Mate in Florida. They want to use Wolbachia, release males of Wolbachia in order to sterilize females and suppress the mosquito population. So, talking about refractory mosquitoes, for example, if a female of mosquitoes has Wolbachia, it's become really refractory to any infection. It looks like it's a vaccination. It looks like we are giving a vaccine, the natural vaccine to the mosquitoes. In this case, they cannot have the virus of dengue or the parasite of malaria, and in this case, we are stopping the transmission of the disease. So this is the program. This is using refractory mosquito harboring Wolbachia is a program from World Mosquito Program. So in our lab, we want to use a strain of Wolbachia called Wolbachia WAU. And this strain is one of the most powerful blocker of viruses within Aedes. And this strain, you can see that this strain can block this dengue virus, Zika virus, and is found in different organs of the mosquitoes at a higher titer. But unfortunately, this strain of Wolbachia is not playing CI. This is a mechanism that makes Wolbachia to spread within population. That means if we want to use this strain of Wolbachia in the field, we need to come with another support in order to help the drive of this Wolbachia in the field. And this is the rationale of our project, because we have one of the powerful Wolbachia strain, and now, unfortunately, it's not playing CI. We need to come with a solution that can support the spread of this strain in the field. That's the idea of my talk. I want you to have that like the main idea of the talk. So we want to do that through entomopathogenic fungi. In our lab, we are working a lot on entomopathogenic fungi. Fungi used to work like chemical insecticide. So they are infecting mosquitoes through contact, And after that, they are going through the different layers of the mosquito cuticle before killing mosquitoes. And in our lab, we have developed different strains of entomopathogenic fungi that we have isolated locally in Burkina, and we have shown that they are really virulent against mosquitoes. They are killing them around 7 days. They are taking around 7 days to kill mosquitoes. And we have also shown that they are really specific to mosquitoes. They are not killing non-target insects like honeybees here, like cockroaches. So when we wanted to come with solutions to support our Wolbachia AU to spread in the field, we went to do literature review, and we have seen a couple of papers showing that in the field there is some Wolbachia. When a mosquitoes, for example, has this Wolbachia, and we infect them with fungi, the fungi cannot kill them. If you recall, I said that some strain of Wolbachia, when they have Wolbachia, some strain of mosquitoes, when they have Wolbachia, some strain of Wolbachia, they become really refractory to any infection. That means, for example, if we want to spread the fungi in the field, as the mosquitoes, when they have Wolbachia, they become really refractory to infection, to any infection. That means we can use the fungi in the field to suppress the natural mosquitoes, and those one who has Wolbachia, that means even the fungi enter in contact with them, they won't die. And we can help, like we can put a positive selection to kill the wild mosquitoes and let drive the Wolbachia ones. So the objective, the objective of our project was to understand what's the mechanism behind this protection when you are working with Wolbachia and the fungi. And also, our project was the first time where we are putting Wolbachia in African mosquito background. And also, we wanted to see if this system can really work in the field. So the design was pretty much simple. We have like, go to the field, make baseline data collecting Aedes mosquitoes in the field, characterize them, bring them to the lab, establish a colony. After having the colony, we make the back crossing of our natural mosquitoes with mosquito which already has Wolbachia in their background, and start to study the co-infection studies. So this is some baseline data. So we have shown that by collecting Aedes mosquitoes in western Burkina, you can see that these mosquitoes are really resistant to insecticide. You are seeing around 60%, 80%. According to the WHO, if the resistance is below 80%, that means that there is a resistance. You cannot really work with this kind of insecticide. That means that the tools that we have in our hands are really failing. We need to come with a new tool. So we establish colonies in the lab. We send the eggs to Glasgow in order to make the back crossing of mosquito which has Wolbachia with our mosquitoes in order to generate the Burkina Faso strain with Wolbachia. And then we made the line colonization. We didn't see any differences when we are comparing the wild mosquitoes with Wolbachia and the mosquitoes our mosquitoes without Wolbachia, and the new line that we put Wolbachia in terms of survival, in terms of hatching rate, and so on. So we did the co-infection study. For the co-infection study, you can see here that when you are infecting mosquitoes which has Wolbachia, you can see that the fungi here, even at the higher dose, which is 10 to the 8 conidia per milliliter, you are not seeing any death. It's like 90% of survival. And with one of our master student, Tatiana, here in the back, she studied what's going on in terms of protection by studying some genes related to the immunity. So she has a poster, and she can explain you better because she knows better than me on that. Okay, yes, this was, thank you. This was really the data that I was showing is under review for publication. We submit the result to the welcome, and really agreed to support us for the next 5 years in order to continue our work with Wolbachia and the fungi in the field. So I have like 5 minutes because I have my timer here also, and I want to finish my presentation with 2-3 slides on COVID. And thank you so much for WANIDA for giving us this great opportunity. We were also skeptical at the beginning of this project, but I can tell you that I was coming here with a lot of confidence because we got a lot of exciting data at the end of the day. So in our team, the idea of our team is to use microbes and insects in order to study evolutionary biology. Why and when a microbe becomes a pathogen for its host. So we can use insects also as a model to study that. So, and during the COVID pandemic, we were asking, a lot of virologists working on that. We are also scientists. What can be our part, our effort on the contribution to fight this pandemic? And we come with this idea that I think it was really a good idea. The idea is that when a mosquito is biting someone who has COVID, it's sure that at some point, when the mosquito is gonna take the blood meal, you're gonna have some antibodies related to the presence of COVID in the mosquito midgut. So the idea here is to use mosquito to study, to track the presence of COVID in the area. Instead of using human, instead of tracking salivary glands or blood to study that. And we have shown through a simple ELISA by tracking like the RBD region or the spike region that we can determine the presence of COVID within mosquitoes and have an idea of the circulation of COVID in some area. For example, entomologists like me, we are used to work in remote area, rural area, where we don't have a lot of infrastructure, PCRs to study that. But we are collecting mosquitoes. Everyone can collect mosquitoes. We just need blood meal and do the ELISA. And we have shown that by working in remote areas, that, for example, over 1,000 samples that we have collected in 12 villages, we have shown that we can have around 28.7. Sorry, I don't have the percentage. This is something that I did really quickly. It's 28% of positive mosquitoes in rural area compared to 45.707% of mosquitoes in the urban area. That means that COVID is really circulating in a rural area of Burkina Faso. And what is really great with that, we want to do what we call ELISA for blood sources. We can know where this COVID came from by doing this ELISA. Maybe the COVID is circulating between cows or monkeys or cats, something like that. So I think I'm really at the end of my presentation. Thank you so much, Peter, and thank you so much, WANIDA, for giving us this really, really great opportunity to showcase what we are doing in our team in iTech MTV in Burkina. Thank you so much.
Thank you, Etienne. We have time for, I guess, one quick question before we have to break very quickly. One quick question over there. Anish.
Thanks for this nice presentation. I have a question concerning the insecticide resistance of EDES that you are working on it. In Burkina Faso, we know that you are working on cotton. So that means that you are using much insecticide for insect control. Did you do what we call biochemical analysis on IDES population of Burkina Faso before starting your experiment? My second question, is it possible that you test your Wolbachia on Anopheles in Burkina Faso, because malaria is more important in West Africa than IDES? If this is okay, I think many people, and particularly National Malaria Control Program, will use this tool to reduce malaria in Africa, and our income will help us to do a lot of things. Thank you.
Yeah, thank you so much, Dr. Ange. Yes, regarding the first question, I don't know if I really get the first one. You said about biocontrol or? Biochemical analysis. No, we didn't do that yet. What we just did was bioassays, and we didn't do any KDR, even PCR for KDR, not yet. But what we have shown is a phenotypical resistance. It's clear that we are not reaching the threshold for WHO. And you are totally right, because the area where we're doing the sampling is really a cotton. They are growing a lot of cotton, cultivating a lot of cotton there. Yeah, that is something that we're going to do for the biochemical, for the molecular resistant experiment regarding the resistance later. So for the second question about Wolbachia, totally agree with you. Malaria is really like the first cause of mortality and hospitalization in Burkina and in West Africa. But what I want to mention here also, for dengue, in Burkina, I'm taking the case of Burkina that I know very well. We have a lot of cases of dengue now, and people are making this confusion by saying palu-dengue, something like that. A lot of people are really, even less than a year ago, we have lost one of our colleagues because the doctor was thinking that it's malaria, and they did the treatment for malaria, and at the end of the day, they realized that it's dengue. But yeah, malaria is the first cause, but dengue also is growing really faster, and we need to come with some solution like Wolbachia for example, if you want to use Wolbachia for SIT, for population suppression. It's the real time now to use this kind of strategy. You don't need to have a lot of cases and a lot of mosquitoes before starting that. The problem with Anopheles is we don't really know yet if Wolbachia is a symbiote of Anopheles. This is an open question in the scientific community working on Wolbachia. And in our lab, I have one postdoc, one of my students, Estelle. She tried to colonize mosquitoes with Anopheles with Wolbachia. But after 2 generations, we are losing all of the mosquitoes. So, yeah, thank you.
Okay, thank you, Etienne. I'm sure there's more conversation that can be had, but we are actually, okay, I'll let Gloria take it, right? We're actually supposed to do a tour of campus, and it's being organized by the staff at the registry, who close at 5 o'clock sharp.
Yeah.
So that means that we need to organize and get on the buses, and it will just be a drive-through of campus and stop at a few key points. But Olivia and Gloria will give you more details and talk about the evening's events.
Okay, so as rightly said, that's what we're going to do from here. So kindly, as you walk out, you grab your snack and then just jump on the bus, and then we'll just go quickly. And then we have a meeting at Fiesta Royal with the students that are on the WANIDA program. So you all head towards Fiesta Royal with the bus, but we'll come back here before we go. So kindly grab your snack, and then we jump on the bus. And then tomorrow we're having the session at Fiesta Royal, not here. Yeah?
Yes.
Okay.
Also to announce to everyone, there's a gala dinner tonight at Fiesta Royale at 8 p.m. and everybody's invited.
Right, so that's it, and we'll continue at WANIDA. Please leave your headset on the seat. Your headset, kindly leave them here. Don't take them away, please. Thank you.
Okay, if anyone has questions online, they can type it in. We'll answer it later. And if you brought a poster here, attention! If you brought a poster here, you have to carry it to Fiesta Royale for tomorrow and Friday.